started pushing that every person is unique, so is every cancer. So cancer is not just one disease or one organ, it's about every individual. So there are 250 types of cancer, and there were 350 genes that are known to contribute to cancer development, and approximately 100,000 mutations have been reported. There's a database called Cosmic Database. If you want, you can search it on Google, COSMIC, which has got all the mutations. It's a catalog of somatic mutations in cancer. C, catalog, O of S, somatic, and M is mutation, I in, and C is cancer. The catalog of somatic mutations in cancer. Somatic mutations means it's mutations that are happening over the life of the person, and it's not inherited. Rather, it's because of the different factors. Like yesterday, we were discussing the effect of alcohol, effect of carcinogens, effect of this and that. So all that is causing mutations. So there are 100,000 mutations known to cause cancer or associated with cancer, but they all zero down to just one unique cancer profile. That's the idea of the personalized medicine. And once again, just to remind that why we are doing it, because cancer is the second leading cause of death in India, not in the world. We are talking about India. This is the second leading cause of death. And that does not just mean death. It's having societal consequences. It has got economical consequences. So it's in the best interest of the nation, best interest of the humanity, that we dedicate ourselves in combating this disease and try to find out something that can help the patients ultimately. And on top of that, in India, we are still a third world country, and that's why we have got limited access to precision medicine in tier 2, 3 cities, let alone not even in the tier 1 cities, this is available. There are very few. And in some cases, even if it is available, it's not affordable. So we have to make it affordable, as well as reaching to the villages and the tier 2, 3 cities. Even bigger problem is our lifestyle changes, which may see 2 million cases by the year 2040. It's not far. We are already in 26 almost. So we have got a big problem coming up, and that's why we need to start preparing ahead. And that's why it needs a affordable indigenous solution. Indigenous solutions means this is for the people of this nation, by the people of this nation, and of the people of this nation. And what is it that we have to deal with is a multifactorial heterogeneous disease that I talked about yesterday. Now, this is what we need, a multidisciplinary team. And I'll show you the examples on how we did this and what are we trying to do such kind of a study based on precision and personalized medicine. Well, request everybody to please mute their microphones, or the administrator can mute everybody. Okay. So multiomics need a multidisciplinary team. And this is a rough framework of how this integration would work. So can you see my pointer?
Yes, sir.
Okay. So we start with a biopsy analysis. Biopsy is the tissue that is taken out from the patient, and it could be a tissue resection as well. We go and do the whole genome sequencing, which is next generation sequencing. And then we also do the gene expression. So this is going to be the genomics. Then gene expression studies or identification of spliced isoforms, which is the RNA level studies. And then the proteomics, the complete protein or metabolite profile, which is going to be proteomics and metabolomics. And blood analysis will give us information about circulating tumor DNA or microRNA, which is also another type of omics that could be done. Once we have all this data, it goes into data integration, which is the work of the computational biologist or the computer engineers. They develop the prediction models, and these models, they are totally in silico. And they provide us some hits or targets that can be identified using these in silico studies. Then the screening of the drug libraries, which are already approved by the FDA, this can be done. Now, here there are options of doing it in silico, or they can be the use of organoids, or they can be use of some other models, preclinical models, where we can screen the drug library and see what is going to work, which will ultimately lead us to get a report for a particular patient that is known as personalized therapy regimen. And we put it in front of the oncologist and tell them that, look, this is what is going to work for this patient, and this is what is not going to work for this patient. So don't bother them with any kind of expensive medicines that are not going to work. So good thing about precision and personalized medicine is that we are not just telling what is going to work for you, we are also telling them what is not going to work for you. So don't bother yourself with trying them. Otherwise, usually the doctors, they say, let's try this, let's try that. No, let's know first and then give them the befitting regimen. So this is how we are trying to do it in the clinic. So it's the same thing. We procure the samples. It goes for pathological assessment. The pathologists look at the tissues and make sure that what we are going to study is tumor and normal cells, so that we can have a clear idea about the tissue that we are dealing with. Now, this will be subjected to molecular studies. This is where the multi-omics is going to happen. The DNA, RNA, protein, metabolites, whatever we can do, depending on the money that we have or with the studies that we can do. And this generates a lot of data. This data is then fed for AI-based model building. Here, we have seen an accelerated phase. As I talked about yesterday, Microsoft and Google, they are all jumping into this bandwagon, and this is a very lucrative career option. If somebody is looking for a career and they're thinking, what should I do? This is the place where we should be seeing the boom and it's highly paid jobs, the data analysis and model building. Once the model is about predictions that how this person disease will be progressing, how fast. Some people, they develop cancer very fast. Some people, they go very slow. What drugs will be working for them? What drugs will not work for them? But this is an in silico model, so we definitely need to test it in the lab, the wet lab, and that needs to be done by using organoids, or if possible, I'll tell you the story about the fly avatars, where Drosophila is used as a preclinical model, and then we can also use the cell line-based model, which is not very accurate, or we can also use the computational prediction models. And after this testing, we can have a reliable report that can go to the oncologist for clinical implementation, right? This is not possible for one person or one lab or one department to do it. So we have got 7 people from different backgrounds. They are coming together to mitigate the effects of cancer. We have got biotechnology, which I am handling, pathology, artificial intelligence, genetics, metabolism, surgery to begin with, and oncology to finish with. This is where the clinical implementation happens, and this is where the sample collection starts. So the first step is the patient sample collection. It goes on in Department of Surgery. So we have prepared some videos for you to show actually how it is being done. So please look at the videos and enjoy it, and then you can note down your questions as well. We'll be happy to take questions as and when you post them on the message box. Swati, can you please play the video for the sample collection?
Yes, sir, on it.
Is it done?
Yes, sir.
Okay, so I will go back to my screen. So Swati, can you tell us something about patient sample collection, because you're already doing it?
Yes, sir,
Because
sure.
there's no voice in that video, so it would be helpful if you can tell us as well.
Okay. So what we do is when we collect the sample, first we go to the OTO. For that, we had to go through a IRB process, that is Indian Review Board. So in Aligarh Muslim University, we have a medical center from where we collect the samples. And first we applied there to get the permission from the review board. And after some interview and some assessments, we got passed through the IRB. And after that, I started going to the JNMC, to the SOT complex. SOT complex is the complex where all the operation takes place. And soon after we started having patients, I went to the operation theater, and the surgeon carries out the operation. And after he removes the tumor, what I do is I take my kit and RNA ladder. So RNA ladder is a kind of compound in which the tissue gets saved for later assessment. So what I do, the tumor that is got removed from, I take one or two sample pieces accordingly, and I put it in the RNA ladder, bring it back to my lab, store it in -20 or -80 refrigerator, and later we carry out the assessment based on the DNA extraction or RNA extraction. And that is how we do the sample collection and further studies. So does that answer your question?
Yes, absolutely. Thank you so much. So this is step number one. We do the patient sample collection where we store the samples. We just try to store it in a way that we can use it later on as well, right? Then this sample goes for pathological assessment, because before we start generating the data, we need to be sure that the tissue that we are dealing with is tumor, and at least 60% of the cells are cancerous. The best approach would be to have 100% cancer cells. And there's a technique called laser capture microdissection by which we can do that. But it's very tedious and it's very time consuming, as well as it costs. It involves a cost. So if somebody has that equipment in their institution, that will be a wonderful thing to do. But if not, then we at least make sure that the pathologists tell us that yes, tumor has got at least 60% cells, and normal is completely normal. This also gives us the staging of the cancer, at what stage is the cancer, which we can later use for any kind of correlation. So here we have got another video from Department of Pathology on how we are doing it. So Swati, can you please play the video, and wherever required, just put your voice there to explain things?
Yes, sir, sure. So right now I explained that the surgeon carries out the operation process, and when the tumor gets removed, this is how they store the tumor and send it to the histopathology lab. So this is formalin fixed specimen.
So this is all about putting the tissue in the wax that can be cut so that they can have sections to study.
Yes, sir. And at that time, no processing was going on, but this rotatory microdome is used to get the sections. And it's like very thin slices of the tissue that they extract. And after that, they study it further. In the slide preparation, what they have done is the slices which they have taken from this previous, wait, I can show you. Yes, the sections which were cut under the rotatory microtome. Using that sections, they prepare the slide, and then they have to dewax it, because after the slide preparation, they get hardened. So to melt it down, they use the warming table.
All right, so this was about the pathology. So the last step here is to have those HNE stain slides. And as I said, can you see my presentation?
Yes,
So,
sir.
yeah, so we have the assessment of the tumors being tumor and normal being normal. So please, all the participants, pay attention to these steps, because we will be soon doing an activity where we will have to play the role of a pathologist or a surgeon. So please pay attention to those things, and we'll be doing this activity soon. So once we are confirmed by the pathologist that cancer tissue is cancer and normal tissue is normal, it comes to our lab, where we do the data generation. We extract the DNA, RNA, protein from the tissue in a single protocol. We homogenize it. We use the kit for extracting all three biomolecules in a single protocol, so that there's not much heterogeneity. And then we use it for sending to the facilities where we can get the next generation sequencing or mass spectrometry for the protein. So, Swati, do you have a slide for this as well?
Yes, yes, sir.
And this also, I think, have some cell culture studies which are used later on, but you can see the video and see how it is being done.
Okay, so I'll first play the cell culture video, then I'll play the DNA isolation video.
I think you can reverse it, because for now we are doing the DNA, RNA, protein. That other one you can play in the validation step.
All right, so I'll start with the DNA isolation.
Yes. So this is the tissue homogenizer that minces the tissue, and then we use a kit for extracting the DNA and RNA that you are seeing in this open dog tubes.
Yes. This tube, like I said, we store the tissue that we collect in the RNA letter. So this is the tissue that we have collected in the RNA letter. We have stored it in the -80 or -20 refrigerator. Now we are going to conduct the DNA isolation. So we have removed the tissue from the refrigerator, and now we are going to process it. For that, first we use the tissue homogenizer to break the tissues into cells and to extract the DNA or RNA. This is a kit. Here we have the Nabzol kit. Here they have all the solvents and reagents for the tissue isolation. And so what happens is the tissue breaks into cell, and when the cell breaks down, the DNA and RNA gets released through the cells. This is a gel electrophoresis setup. So what we do, once we isolate the DNA, we add the samples into these wells. If you can see it, these are the wells. So we mix the DNA with loading dye, and after that we load the entire mixture to these wells, and then run the gel from positive to negative. Okay, so this shows that where I have written DNA, it means that we have taken one control. It could be a DNA ladder or some known DNA sample. This sample is what we have used recently. I mean, the tissue that we have extracted. So after the gel is run, this is what we observe. So these highlighted areas show that the DNA has been extracted.
So people from molecular biology background will appreciate that the sodium bromide stains the DNA, and that's what we are seeing on the gel, right? So once we have got this DNA RNA, we send it for next generation sequencing, and they provide us with the data. Now this data goes to the AI department, artificial intelligence. We have got a center for artificial intelligence where they work on this data. And this is the toughest step as of now, but it's getting easier with the help of the supercomputers, with the help of these advancements in the AI. This is called data integration, and they help us with building prediction models as well as finding out the targets. Remember that this is just an in silico exercise. I'm repeating that because there's a lot of errors that might come into this because based on the data quality that we are getting. Right. So we'll show you another video. This video does not have exactly for the cancer, but the students have developed some models which could be used for doing the cancer target identification as well. So Swati, can you play the next video, please?
Sure, sir.
This is overview of our AI lab. We do have 9 cell systems here and a workstation. We do also have.
You can skip that, all the setup and everything.
Okay.
And can go straight to the work.
Captures important. To address this model focuses on skill design classification using a multi-model deep learning framework. Skin cancer cases are increasing rapidly, and early diagnosis plays a critical role in improving survival rates. However, traditional diagnosis depends heavily on expert dermatologists and visual inspection, which may not be always accessible. To address this, we used HAM 1000 dataset, which contains reference 40 images along with patient metadata such as age, gender, and recent location. We processed image data using DepNet 1-to-1 and a powerful convolution network and tabular patient data using DepNet, which effectively captures important clinical features through attention mechanism. As shown in the confusion matrix, DepNet strongly perform on image-based classification while DepNet perform reliable predictions using metadata. These results indicate that both modalities contribute valuable information, and combining them can further improve performance. In conclusion, my project demonstrates that multimodal learning enhances classification, accuracy, and reliability compared to single source models. In the next phase, we plan to apply reason segmentation and advanced reason techniques to build a more robust and clinical useful diagnostic system. Thank you.
Implementation for diabetic patient. I have used 5 machine learning models and 1 deep learning model for direct recommendation. You can see random forest, SVM, gradient PhD, XGBoost, AdaBoost, and multi-layer perceptron. I have used user-generated data from many clinics. I have collected it, and I have used Indian diet recommendation data for recommendation. Here we can see age, gender, weight, height, current glucose, A1AC, and dietary preference is given by user. And breakfast meal for breakfast, lunch, evening, and dinner is given by the models. And the total whole day calorie and carbs, protein, fibers are divided according to it. And the total achieved calorie we can see here is a little bit different from the target calorie.
So this is actually personalized medicine. That for every person, we are talking about a different calorific need. So this is something that we can use in precision medicine. And the previous slide that you saw was about the multimodal models, which is about integrating the multi-omics. Let's see what's the next one. It's about Alzheimer's using machine learning.
For Alzheimer's disease analysis, combining brain MRI images and clinical community scores to improve diagnostic accuracy and reduce severity estimation. Alzheimer's disease is a progressive neurological disorder, and early and reliable diagnosis is essential for effective clinical intervention. Here, we use the ADNI dataset, which consists of structural MRI images along with 4 clinical assessment scores: ADA, MMSE, CDR-SB, and SAQ. For feature extraction, Resnick 50 was used to capture discriminative spatial patterns from MRI images related to brain atrophy. The cognitive scores were processed using a bidirectional LSTM, which effectively models relationships between multiple clinical assessments. The proposed framework performs 2 tasks simultaneously. The first task is multi-class classification, where subjects are classified into Alzheimer's disease, microcognitive impairment, and normal control. The second task is integration, where the model predicts the 4 cognitive scores, providing a continuous measure of disease severity. Also, here are the results. The results show that our model achieves 83% classification accuracy, and confusion matrix indicates reliable differentiation between disease stages. Also, the multitask learning approach also improves the regression performance by sharing representation across tasks. So, in conclusion, the project demonstrated that combining multimodal data with multitask learning leads to robust and meaningful Alzheimer's disease detection. Thanks.
This was also a multi-model data where they're taking images, and we are not yet going into that complexity that you can also integrate images, and you can also integrate the blood reports into this data analysis platform. And now it's being done. But I'm trying to keep it simple because we have got a diverse audience. So we are just talking about DNA, RNA, protein, and metabolites, but we can integrate other information for the patient as well. Next is, once we are from these prediction models, we are given the targets, we go for target validation. And it happens in a molecular genetics lab in zoology, or metabolism lab in endocrinology. And then it's also being done in our lab, where we validate the models using cell culture. And we are also going to develop the organoids, which will be giving a better recapitulation of the patient. Now, organoids are of 2 types. One is like from the cell lines, and the other one is patient-derived organoids, which means every patient's tumor would be prepared in the form of organoids so that we have got maximum similarity from the patient itself. So that's what we are trying to do and establish in our lab. So our next video will show you about the cell culture and how we are playing with the cells to identify those targets and validate them. Adi, can you please play the next video? Swati, are you there?
Yes, sir. Yes, sir. I'm on it.
So please feel free to post your questions in the message box. We'll be happy to answer them as we move around. So let us keep it very interactive and enjoyable experience. So this is something that is going to be the deciding phase, because we know a lot from the prediction models, but we have to test it, and then we have to give it to the clinicians. So this is a crucial phase in the entire setup, that because we will be telling the clinicians that, look, this is going to work and this is not going to work. This is the right target against which you should be giving the drugs, and this is the target that should be avoided. So this is a critical phase. Now, in all this thing, one important aspect is the time. We cannot work on an open-ended scheme, because patient is there, and we have to present to them the complete report in a time frame that can be useful. So far, it is this is also a challenge, that we can identify the targets and give them the report in time. Because starting when the surgery was done and the tissue was taken out, and when we present the report to the clinician, this time, if it is too long, then it is useless. By that time, the patient may die. So we try to minimize this time, and so far, different models have got different time frame, but it still takes 6 to 7 months. That is another challenge that we need to address in speeding up. Yeah, so you're ready now. You can please give a voice to it as well, your own voice of what they're doing. Okay, I will do that. So here, this is the biosafety cabinet where we are maintaining a sterile environment for the cells to grow, because we want only the cells to grow that we want to test with, not all the cells. So we can have cells from different organs like colorectal cancer, breast cancer, or blood cancer, or different lung cancer, different other types of cancer. So each of them have got immortalized cell lines, and we maintain them in these cell lines. They need a special media, which you see here in the red, and this media is given to them as their food. So once we take out these cells, which we store in -80 or liquid nitrogen, this is what we are trying to revive them. We will take the cells and put them in a flask, the green capped plastic container that you're seeing, and then we will be putting the media into it, which will work as food so that they can grow. Once they grow enough, we will be able to see them and test them and use the drugs against them. Here, I think she's centrifuging it so that we can give it a wash before we put them in the flasks. So now these cells will be.
अपने लास्ट बता रहे हैं।
Can you please mute your microphones? Sure, some of you have already done it, but we have got, as I said, diverse audience, so we want to make sure that everybody is on the same page. Yeah, this is the media, so we will be adding the media and resuspend the cells. This whole process has to be very gentle so that the cells are happy and they don't die. Because if they die during this revival process, they were sitting in the -80 or -196 degrees Celsius for a long time. Now they're coming back and they are being given the chance to survive. This process has to be very gentle so that most of them can survive. Still, not all of them survive, only a few of them survive. Yes, I'm seeing that. Yeah, they go into because they use a cryopreservation agent, which is usually DMSO. And this cryopreservation agent does not allow them to freeze. They are at this temperature, but they don't freeze. Plus, we also give them very high rich nutrients. So they just sleep, but they don't die. Yeah, if you can put yourself in a bag of DMSO, which is impossible. These cells, they can lower down the temperature and survive. But human beings cannot, because they have got multiple inlets and outlets, which will be absorbing all those things, and that will be fatal. Now she has kept it in the green flask, which will be the home for these cells. And we need to keep changing this home every 2-3 times we passage them. This process called passaging. And cells cannot just keep going over the time. Rather, we have to make sure that after certain passages, they are freeze back so that we can, it's like the life of the cell. Although they're immortal, but still they change over the time. This is the microscope we are using. So these are the cells that, while they are round, as they have been just recently taken out from the minus 80 degrees Celsius, so they are round, which is showing that they're stressed. But usually they become nice spindle shaped, and they also are adherent cell lines, which means they will stick to the surface of the flask. I think after 24 hours, now you can see that this is how they are. This is their normal morphology, and they're growing. And they grow in colonies. Some of them they grow individually, and for growing also we have to provide them a nice home, which has got controlled CO2, 5% CO2, and 95% humidity, as well as a completely sterile environment. This is the incubator. So we have to really take care of these cells in a very nice way. And once we have to do any experiment, we need to know how many cells are we dealing with. For that, we use hemocytometer. Although now we have very advanced equipment which can do the automated cell counting, but we are still doing it traditional way. Hemocytometer is pretty accurate, and this is used for measuring the number of cells that we are going to deal with. So we stain it with a dye so that we can know it's trypan blue, which can tell us whether the cells are live or dead. And this will, so out of the cells that we're counting, we also need to know which one of them are live and which one of them are dead. So live cells will be able to take up the dye, and we count them like that. And the dead cells will not be able to see it. So you will see in the microscope that the cells are live or dead. We can also tell it about this from the morphology. This is how we count the cells. The 4 squares that you're seeing at the corners, the B, C, D, and E, they are mostly used for counting the cells. We are not going into details of it, but then this is an assay that we do for measuring the effect of the drugs on the cells, which is called MTT, or there's a modified version called MTS assay. For that, we need to put the cells in a 96-well plate, equal number of cells. And once we put the cells there, we can either put the drug along with the cells at the same time, or we allow the cells to adhere for 24 hours. And the next day, we can add the drugs. So you can imagine we can try 96 different combinations of drugs and know which one is going to kill the cells and which one of them is not going to kill the cells and by what percentage. So here's a different color, then we read the absorbance so that we know which one of them are dead and which one of them are not. So this assay is a very basic assay which tells us about the cell viability, that how many cells survived, how many cells got killed. So this can tell us about the effect of the cells, and since it is quantitative, we can also find out the IC50 values, the value, the concentration of the drug at which the 50% of the cells are dead. This also tells us about the potency of the drug. So this is a plate reader where the absorbance will be read for all the wells. It's a simple absorbance reader, and different reagents have got different wavelengths at which they read. So you can read it at 595 nanometers here. There are many other steps about calibration, etc., which we are not going to talk about. But ultimately, what you get is the effect of drugs or any other material that you want to test on the cells. So we got the targets from the in silico people, from the AI people, and now we are testing those targets on these cells, right? Ideally, it should be the same cells that we extract from the patients. For that, as I told you, we'll be doing the organoids, which can tell us about the exact results from the same patient. But here, these are the cell lines that have been maintained for years, and they also can give us some idea about the targets itself. All right, I think these are the numerical values that you can use for plotting your data for knowing what is the drug concentration that is going to kill the cell, 50% of the cells, and so you can tell the clinicians about the concentration of the drugs that need to be used in physiological conditions. Okay. I think that's it. Sadi, we can. We're already 11:50. Good. So this was the target validation step that we do in our lab, cancer nanomedicine lab. And then whatever report we generate, that goes to the oncology, and there's a separate department of oncology. So you can see how different expertise, different specialties we are working in sync to find out the precise and personalized therapy for a cancer patient. So we have another video from the oncology department, how they're dealing with the patients. And Swati, can you play it, please? Skip those portions which are not relevant. I mean, okay, sure. I'll tell you. Sure. So you see here how when we give these reports to the oncology people, how they are dealing with the patients. This is the Department of Radiotherapy, which is taking care of all. This is not just about radiation, rather this is about the dealing with the patients themselves. I think you can skip this and come patient waiting area. Here, this is the patient waiting area. Okay. This is what they're doing, the radiotherapy is going on. They also have got a microbiome cancer research unit, so they're also studying the effect of microbiome. This is another omics that we can add, but I'm not telling you just for the sake of simplicity. We're not adding more omics to it, but we can also have the data from the patient's gut microbiome, the bacteria that are living there and the microorganisms that are living there. So they have got lab setup that you just saw, maintaining the sterile environment, and then accelerate that. So we also have a National Cancer Registry program, which means every patient's recorded so that we have the data about the effect of what we are doing. So we are giving personalized medicine, precision medicine, and what is the effect of doing this actually in the patients. That's the cancer OPD, where the patients are being seen. The trained oncologists, they are dealing with. The final call rests with these oncologists. They have to decide either alone or with the tumor board that which therapy is to be given to the patients. Because we are seeing only the omics and other data, they have to see the patient itself, whether the patient is able to take these medicines or not. So the final buck stops at the oncology unit for administering there's also a palliative care. Palliative care means the people in the advanced stages, we just need to work on reducing the pain. So that's the palliative, and there's separate organ-wise clinics, head and neck, separately gastrointestinal tract or the lung, and skip that X-ray simulator room. Yeah. Then population-based cancer registry, again, this is important that we need to know how many cases are coming and what is the effect of our therapy on the outcome in terms of the survival statistics. Accelerate that. This is daycare ward, that's fine. So these are all the medical facilities we have here in JNMC to implement the precision and personalized medicine. All right, fair enough. This is how the patients are being taken care of. Good. So now here, if the precision and personalized medicine works, we will see better happy patients coming out of the clinic and better survival rates. If not, then it will be a test for us to go back and test again. This is also a place where we get the feedback from the oncologist that what are the problems that you're facing so that we can go back to the lab and work on it so that we can refine our reports, right? So this is the setup that we have, and we call it Cancer Nanomedicine Consortium. So this I've shown you earlier. There are different expertise working together. Keep this in your mind because we are going to do the activity where you'll be reading these terms. So this is how we are hoping that this is how we are hoping the flow of work will be. So we integrate the omics. That's only a part of the starting story. We go on to understand the patients in an individualized manner. We call it molecular classification. So we classify the patients about which drug is going to work on which class of patients. And then we characterize the individual based on the molecular signature. In the preclinical models that you saw in the cell culture, we are going to test the drugs and tell which one is going to work and which one is not going to work. And that leads us to N is equal to 1 trials. If you remember yesterday, I was talking about the population level trials, where you see the statistics and then you say whether this drug is working or not. That is an old, or that way does not work. Rather, N is equal to 1 trial means there's a trial for 1 patient. So we are just working on 1 patient. We're trying to give them the best treatment that's possible. All right, so we have got this poll here already going on. So these are the 2 approaches which we saw, that multi-omics for finding the targets versus single omics for finding the target. Let's see how the poll was about traditional therapy and multi-omics based therapy. I see how many of them have responded. They're just 17 votes. Come on, everybody. We are 82 participants. For the poll, you can go at the bottom right corner. You will see the dots, these 9 dots. And click on them, you will find the poll. Click on them and tell us what do you favor for cancer therapy? 17 votes. Yes, please. I think all of you can vote, 82 participants, so that we can have a fair idea of making teams now, which is the next step. So while you're doing the polling, I just want to remind you again, these are the traditional therapy approaches in cancer: chemotherapy, targeted therapy, surgery, radiation, and recently the immunotherapy. We put them all under traditional therapy. And this other class is about the personalized and precision medicine, where we identify the targets using multi-omics, we characterize an individual, and then we give the right therapy to the right patient. This is stuff that I've talked about in the beginning. Yes, having the right patient, the personalized medicine, and the precision medicine for successful outcome. So these are the 2 concepts that we are going to discuss: traditional therapy versus precision and personalized medicine. So now the activity will be, I'll be making the break rooms. And break rooms will have, it will be either as a consortium team or a traditional team. So each team has to choose a type of cancer. You can take up any organ type of cancer. And if you are in the consortium team, you have to discuss the multi-omics based precision and personalized medicine approach. You saw all those steps just now, from surgery to oncology. You discuss all the steps and prepare your presentation. And the traditional team will also take up, either they're taking chemotherapy or they're taking radiotherapy or they're taking the immunotherapy. And then you will have some time with you. And then one of you from each team will be presenting. Is that fair? Okay, so I will make the break rooms. We have got 82 participants. Let us make 8 teams. 10 each. Okay, so let's see, break rooms. So again, these are available here. Set up break rooms. I will make 10 rooms here. We'll be randomly assigning you, or we can just make 8 rooms. That's enough. So how many of you would like half and half? Let's do it half and half. So you have to play the role of a surgeon or a pathologist or this and that. And you have to discuss among yourself and then present your case that why do you think this is the best strategy for treating these cancer patients, right? So I will make these rooms traditional therapy. Let me see the poll. I think most of you are favoring multi-omics. So let me make 3:2. So out of 8 rooms, we can have 5 for the multi-omics, traditional therapy, and 3 for the traditional therapy. So 1 traditional therapy, 1 precision medicine. This one also can be precision medicine. This one can be traditional approach. Approach. Normalized. It's the same as precision. So I'll soon open the rooms, and you will find I've shuffled. There's no way that I can know whom of you want to go and which room. So I've just used automated shuffle, and you will find yourself. So introduce yourself, and this is another skill that you need to develop in talking to each other, cross-disciplinary. See, like we talk, I don't know how a surgeon works. A surgeon doesn't know what molecular biology is, but we talk. We don't know how the AI people work, the model building and all, but we talk and we understand each other and we try to develop the solution. Khalida, just to wait for a moment. I'm just making the rooms, and soon you'll find yourself assigned the rooms. Okay, so I've opened the rooms. Can you see that? So make sure that you're not more than 10 in one. Swati, you are in one traditional therapy room. And Sayeda is also in the same room. Can you change the room, please? So we can have some moderation, and I'll also join some of the rooms. So just see if some room has got a lot of people. We can balance it out. Ahmed Saeed, Alpa Singh, Anam Riaz, Anzarul Haq. Can you please join the rooms? If there's any problem, let us know. Dr. Murad, Dr. Tohasif Chaudhary.
I'm not sure if it is with me, but if I'm joining the room, I'm being logged out from this meeting.
Yes, that's fine. This is what it is meant for.
Okay.
You have to go into the room, not the main lobby. So you're not, you're going to the rooms, and so you'll be logged out from the main. And then we will rejoin after 30 minutes or 20 minutes, let's say 20 minutes, so that we can have presentations. Is that clear? Dr. Murad, Aparna, Arfin Hussain, please join the room so that I lost the room connection. Okay. How come? Let me see this one.
Hello everyone. This is the consortium room, right? When anybody speaks.
May I request the people in the main room to go in the breakout rooms? Because some of the rooms are empty. Aparna, Anzar al Haq, Asna Khan, Atul Kumar, Murad Hussain, Fatima. On your mics are unmuted, so you can just go in the room and start preparing the plan.
So sorry, sir, for coming here. But hello,
Yes, yes, go ahead.
sir, in the background room, in the breakout room, no one is
Mhm.
actually talking. Like they are not even interested. I guess like I've tried, but no one is there opening their mic
Okay,
and speaking.
okay. That's also something that is a problem in the digital age. That right. So we can't do anything about it. So let's see.
Yes, that's why I thought this is the breakout room, so we were talking. So sorry, sir.
But it's still, you are in which group so that you can present. We'll just be able to call a couple, 2, 3
Uh,
people from the whole group.
okay.
So,
Sir, were in the multi-omics section.
okay. So
Yes.
if there's nobody, then just prepare by yourself.
Okay. Sir, I had a question.
Uh-huh.
Uh, is this right? Like, you need us to explain all those topics, like the
No, no,
topics
no.
for
Just
precision medicine and clinic.
no. Just imagine a patient is coming to you, and what will you do based on precision personalized approach to treat? It's more like what you have learned so far. We want to see. We want to hear it in your words.
Okay. As an individual, I can speak with all
Yeah,
the steps.
that's fine. It's better if you do the group consultation. That will be better. But if not, then
Okay.
that's another option. Let's see. So Khalida
So,
Siraj, Juhi Gupta, were you not able to join Mahi Patel, any group, or was there any technical problem? Mustafa? No one speaking in your group. Okay. So were you speaking? So Aparna is there in your room or no? So Mustafa, Aparna, you can connect and see. You can at least, both of you have the same problem. Nobody's speaking.
Uh, I'm Dr. Juhi. In which group I am? Actually, I joined today very late.
Yes, Dr. Juhi, we have discussed so far the multi-omics approach for precision and personalized medicine. So we were doing this activity for imagining a patient coming to your clinic or your lab, and how you can develop a precision medicine based approach or traditional therapy based approach to treat that patient. So whatever you have learned so far, we wanted to hear it in your words. Did you get the concept of multi-omics and how we are doing all the steps of surgery and pathology and target validation and data analysis and all that?
Okay.
So there are breakout rooms. If you want to join those breakout rooms, you can take up.
Yeah, it's better if I join some group.
Yeah, there are some breakout rooms. If you go at the bottom, and you'll find the meeting tools, those 9 dots. And if you click on that, you'll find the breakout rooms, and you can join any one of them. They are categorized as precision and personalized or traditional therapy.
Okay.
So we'll end these rooms in a few minutes by 12:30. I think there is not much time. We'll end these rooms and then we'll ask people to present. Niloufar Bano, Saiful Zafar, Sana Jamil, Kulsum, Muskan Khaliq. If you want, even you can use the main room for talking to each other, and because all of you are now unmuted, you can talk about choosing a precision or personalized medicine approach or a traditional therapy approach.
Multimonic, I have just polled for that.
Okay. All right, so I have closed all the rooms. So everybody will be here in 30 seconds. Yeah, I think people are coming in. There's still some people in some groups hanging on. So once they are back, let's have our first presenter. Who's ready to present? We can volunteer. Or Aparna, would you be willing to be the first presenter?
No, sir, not yet.
You're not yet ready? Who is ready to present? Because we have to conclude the session at 1. I hope we had more time to do this, right? So anybody from the participants would like to present their strategy based on precision and personalized medicine approach or traditional approach?
Yeah, sir.
Ilma Rafat,
Yeah, sir. Hi, sir.
you can use if you have some slides prepared, fine. Otherwise, you can just speak, and that should be fine as well. So what you have to tell us is what is the patient, what kind of cancer they have, what approach you are going to use it, how you are going to use it, and probably why you are going to use it. Okay.
Okay, sir. Sir, I can not see the rooms and the participants that we have
Yeah, I closed already. I closed that
yeah,
already. The time is up.
yeah.
So now it's time for presenting.
So we have divided some parts to the matlab our groups. So I have taken the presenting part. So
Okay.
I want to know my group members and their names and their parts. I have lost that chat box.
Okay. Then just present. I think it's not difficult to
Yeah, sir.
retrieve them because this was randomly done by the computer,
Yeah,
the
sir.
group assignment. So I have no records for that. So you can go ahead, whatever you discussed, and present your patient. Yes, Farha, you can be after this. So you have to present your patient, you have to present your approach, and you have to tell us how you're going to do it, why you are going to do it. Just for these four things. If you keep it simple and fast, it will be better. Go ahead.
Okay, sir. Sir, I'm presenting my own part.
Yes, Ilma.
So let's start with a brief introduction of mine. That I am a PG first year scholar in the Unani medicine from
Mhm.
NRUM Hyderabad. So I will like to treat my patient, lung cancer patient, with the Unani approach. So according to Unani philosophy, we call the cancer as a Saudabi Amrad, that Amrad Saudabi according to our pathy. So it occurs into the part of the body according to our literature in the pathy. What are our beliefs? So not been performed in the patients as it is the don't have any evidence-based research right now. So we cannot apply the I cannot apply the surgery part in the lung cancer patient, but I can treat the patient as a supportive management, like the post-surgery management with the Unani therapy. Am I audible?
Yes, Ilma. Go ahead. Once you complete, then we'll be discussing.
Okay, sir. So according to our Unani, we have a therapeutic approach, like we have a dietary approach, the drug therapy, and the regimenal therapy. Dietary therapy with the support of the dietary advancements, and drugs with our herbal formulations or the polyherbal formulations, and with the regimenal therapy like we can have pain management, the Bukhur therapy, the hijama cupping therapy, the exercises, the dalak that we call that the regimenal therapy. So post-surgery management will be like we can give the patient the immunomodulators. We have a very good immunomodulators in our Unani therapy. Like we can give a Khamira Margarit Khas, the compound formulation, the name of our compound formulation is this. Like we can give the liver tonic that will strengthen up the liver and detoxify the toxins, like a Jigreen, Jigrol, that are the names. In the pain management, we will go give the Majun Suranjan, Habbe Azgan. We have research going on and we have a very good effects in the management of the symptoms and reliefs, and the post-operative assessment of the patients are really good. So this is the whole approach in at a very glimpse I have shared.
Yes, Ilma, very nice that you are using your traditional method. But don't you think it's just hit and trial? You will give
No.
because there's no evidence of which target are you going to identify, and what
No,
will
sir.
be the
No, sir.
Good.
No, sir. We have the compound formulation in the given in the
what is the
classical
target for
test,
that
and
formulation?
yeah, I am telling you that I am coming on that part.
Mhm.
So the for compound formulations that we are using have the clinical evidence-based researches, and we have published journals and published texts
That's fine.
that
So tell
it
me the
have,
target.
yeah,
Tell me the target. What is the target of your preparation?
that we will target that as I have said that I'm not going to treat the cancer, the lung cancer.
Mhm.
But I will give the Unani medicine as a supportive treatment, as a post
So
therapy.
it's mostly symptomatic,
Yeah,
symptomatic treatment, right? So you'll be treating
yeah,
the symptoms. Okay.
yeah, yeah.
All right.
That is
So
not a direct.
thank you
Sorry.
so much. And we will move on to the next one. Farha, I think you
Okay,
volunteered to be the next one.
okay. Thank you, sir.
Okay,
Thank
doctor.
you, Ilma. You brought a very good aspect to the therapy. Farha, are
Okay,
you
hello.
ready?
Yes, doctor, I'm ready.
Okay. I'm
I have.
giving you a timer with 5 minutes. So please stick if it's possible. Otherwise, it's fine. Go ahead.
Okay, hello everyone. My name is Farah Salida. Actually, I'm studying in University Saint Louis. So my target is usually we are this cancer cell. So well, I'm taking its age is 2 years. He's a female and caused by stage 3 cancer. So obviously by the like hormone status estrogen is positive PR plus PR2. So what treatment plan? Basically, first we have to do the surgical management, that is lipotomy with sentinel node biopsy. If the chemotherapies like doxorubicin or plus cyclophosphamide followed by the pathologics. Radiotherapy we are using, like for example, external beam radiotherapy and to the breast tissue. So for mine is clinical implementation is multidisciplinary team, like involvement of the medical oncologist, surgical oncologist, radiation oncologist, and clinical pharmacist, and oncology nurses. So what we do with breast cancer, we have monitoring the chemotherapy related adverse drugs reactions. Secondly, we can regularly imagining and laboratory investigation. Third, we can and evidence monitoring. Now, what we do the expected outcome, we don't know that, like reduce risk of reoccurrence. Secondly, improve the disease-free survivals and improve overall survivals and better quality of life with supportive care. So based on PR that like in we can say the conclusion, traditional therapies remain as the backbone of the breast cancer management, offering effective and clinically proven outcome when implemented systemically within multidisciplinary clinical care. So if we see the normally like patients from uh, treatment like traditional therapy for breast cancer, first we must to diagnose biopsy test. We can see the ER and PR receptors. Then we have a staging like TNM classifications. Then primary treatment. First we have to go for the surgery and chemotherapy. Like we use the doxorubicin, most medication used for the breast cancer. If the resistance, we can change the anti-cancer drug. Then third is radiotherapy. Radiotherapy, like fourth stage, we can go for the adjuvant therapy, like hormonal therapy. We can use the tamoxifen and follow up and monitoring, like regular imagining test, like blood test. So mine is
Good, Farha. I think that's it. You did it well in time. My question would be, I know that we are already doing it traditional way, but do you think this is improving anything in terms of the mortality that we are seeing? Because these are the targets that have been identified based on incomplete evidence, incomplete pictures. HER2 was found based on the genetic analyses and the availability on the surface. So we are not progressing on that. So people are still dying with breast cancer. So we have to think differently in using multi-omics. If it was treating, then it's fine. Then I wouldn't have contested it. But the reality is that people are still dying of these cancers, and the recalcitrant disease is also common. So think about using a different approach, and that could be perhaps precision personalized medicine using multi-omics.
Can
Right,
you say,
Aziz?
Doctor, something?
Uh,
Reg-
I'm sorry, we can discuss it later on. Let's have Homa.
okay.
You are the next one to speak.
Okay, thank you.
Thank you for. Homa, you're there?
Hello, sir.
Yeah.
Yes, sir. My name is Dr. Huma, and I am first year PG student. And, sir, can you hear me, sir?
Where? Yes. What's your background?
Yes, sir. My name is Dr. Huma, and I am first year medical MD student from Unani Medicine at NRIM SG Hyderabad
Okay.
in my batch.
All right. So I think Unani medicine is going to dominate today. Anyways, go on. Your timer has started.
Yes, sir. I'm really sorry. I'm really sorry, sir. Also, I've chosen the topic as breast cancer. And as we know, breast cancer is heterogeneous with multiple subtypes. And my approach is not traditional approach. I'll be going with multi-omics approach. So it will integrate genomics, proteomics, metabolomics, and to understand the cancer complexity. So, in genomics, there are various tools to detect mutations like BRCA1 to TP53, HER2 amplifications. And proteomics, we can identify protein biomarkers like HER2, ER, PR levels, and then we can predict therapy response. And in metabolomics, we can study metabolic reprogramming like lipid metabolism alterations, potential drug targets, and all. So, how we can be precise medicine in breast cancer. So, we can even use molecular profiling to tailor therapy. Like, for example, if we have to target like BRCA mutation tumors, we can give PARP inhibitors. If we are targeting HER2 plus tumors, we can give pertuzumab or hormonal receptor positive like tamoxifen aromatase inhibitors we can give. So, we have to focus on individual patients, like tumor biology, genetics, lifestyle, and environment. So we can use various tools here, like liquid biopsy, to monitor therapy response and detect early response. So, integration of multi-omics plus clinical data that allows personalized treatment plans. And when we talk about the clinical impact, so we can predict therapy response, detect resistance early. So if we can detect resistance early, we can modify the therapy. Also, we can improve the survival and quality of life. So my key message would be like in breast cancer, combining multi-omics approaches with precision and personalized medicine allows us to move from one size fits all therapy to patient specific treatment strategies, improving outcomes and minimizing side effects. Thank you.
Thank you, Dr. Huma. It was wonderful. I agree that you are for the precision personalized medicine using multi-omics. And here's an important point that there's a lot of drugs that are available in the Unani or Ayurvedic system, and those drugs can be tried. Once you have the right target based on your multi-omics study, you can use those drugs for testing that this patient is going to be getting benefited from this majun or this something, this concoction, or this patient will not. So you can do some preclinical testing and then give it to them. Additionally, you have mentioned about the diagnosis and biomarkers. Yes, we'll talk about that later as well. That's another important strategy in doing personalized medicine. All right, thank you so much. I think we have next Dr. Padmavati Ganeshan. Dr. Padmavati, are you there?
Yes, sir. Yes, sir, I'm here.
Okay. So let me just reactivate your timer. And yes, timer is on the right top hand corner. Please start.
So I'm Dr. Padmavati Ganeshan. I'm working as an assistant professor in the Department of School of Life Sciences in Present University, Chennai. So in our group, we decided on the cancer, so which cancer we are going to talk about. So we decided on lung cancer. So before we decided on the topics and everything, we lost the group. So I'll be presenting on behalf of them. So basically, I focused on lung adenocancers. So lung adenocarcinoma, it's like the general drugs used for these therapies are platinum-based drugs, which are like targeting the DNA synthesis and overall process, which might affect the other cells, normal cells as well. And also there are specific mutations like EGFR mutations or KRAS mutation between one patient to another patient. There are different specific mutations which will change the outcome of the treatment option, the traditional treatment option. So my approach would be to use the multi-omics approach to find out what type of mutations present in patients, and then according to them we can do the targeted therapies so that the treatment option or treatment will be very effective. The therapy will be very effective for the particular patients. So basically, like we have seen in the lecture, we will first start with the surgical procedure. We will do the surgical removal and we will go for the DNA RNA excretion, and we will find out the genomic analysis to find out the specific, for example, in case of KRAS mutation, what type of KRAS mutation is present in that particular patient sample. And once we identified the patient, particular KRAS mutations, we can just proceed with the in vitro validation. And if we have the specific compounds, which, for example, KRAS G12C mutation, there are FDA approved compounds like Lunocrac-KRAS and KRAS. So like these kind of compounds we can use. If there are some other specific mutations, if we are able to identify through other computational approach like molecular studies and all, if we are able to identify the specific components which could target this particular KRAS mutations, and if we can use those compounds, it can be either plant-derived compounds or maybe a synthetic compounds. If we can use those compounds for the treatment of the patients, it will be more effective and very personalized to that individual which carries that particular KRAS mutation. So that would be my approach for the treatment of patients with lung adenocarcinoma.
I think that's excellent, but this is what is standard of care as well. So that's what we say, that we have got some standard of care procedures that are in place, and most of them are for all the patients. This is what we call as the one-size-fits-all approach. But now we are seeing even lung cancer and tract fusion. This is for non-small cell lung carcinoma. So we can do some prior assessment for these patients and see some preclinical testing. Unfortunately, this involves a lot of money, and patients are not able to afford that, not even at one sequencing. So that is something we need to think about as well, that how can we make it more affordable for the patients. In US, they have got a lot of things now covered through the insurance schemes. If we have got similar, I don't know if you have got that mechanism in your hospital to cover for every patient sequencing. Is that possible in your hospital?
No, sir, we are not. I'm not working in a hospital. It's an educational institute.
Okay.
Yeah.
Thank you. We have to think about these as well, the affordability issues.
Yeah,
So
true,
for
sir.
that,
But
people are making some channels.
in Adyar Cancer Institute, in recent days, they have developed this kind of methodologies, genomic approach, multi-omic approach, how we can approach the cancer patients with these things. So those facilities are available in Adyar Cancer Institutes. They are doing some research based on that, but it is still in the research lab level, so they haven't applied on the patients yet.
Yes, I think Dr. Asim would also tell you some success stories that came out from his lab and went on for now they are in the clinic. So that will be motivating for us and to do something here in our country as well.
Yes, sir.
All right. Thank you so much. Next, we have got Dr. Sandeepan with us. Is
Hi,
that
Dr.
right?
Aziz. I'm not Dr. Sandeepan yet, but I'm just a master's student right now.
I'm motivating you to
Uh,
be one.
Okay,
All right.
thanks
So
for
your
the
time will start
wishes.
now.
Okay, thank you. I would like to present the pointers presented by Dr. Amar Aziz and also Dr. Khalida Siraj and Dr. Fatima as well.
Can
So
you
I'll
introduce
like
yourself,
to
please?
myself Sandeepan Acharji. I did my master's in biotechnology from Himachal Pradesh University. And in my dissertation project, I developed a hydrogel-based drug delivery system for cancers. So I worked on rhabdosarcoma cells, but I think I need a much more precision work for that. So I'm just looking for PhD positions right now. So currently, I'm just talking. I would like to present my topic, that is bladder cancers, as decided by a team. So, as Khalida presented for clinical aspect, that a person comes with a hematuria. So, the the patient is having bladder cancer. So, first of all, the treatment on the traditional side, we see that in newer stages, or stage 0, stage 1, or stage 2, we go for QRBT surgeries, where we just extract out the tumor cells, and we just prevent the recurrences through BCG treatment. And BCG is basically immunotherapy regimen given in every 6 week span. So this thing is not much developed in last 2 decades, as we can see. But also we were talking about how we could develop precision medicine. So as Dr. Amar Aziz said right now, that we can look for the cell types, which cell is infiltrating, which cell is being responsible for the cancer type. We can also look for the biopsies, as, like we have discussed, that if we look for the biopsies, the cell types or the cell aggregation, how it is infiltrating to the other sites, we can thereby decide the regimen of the treatment. For instance, if it has metastasized on a huge scale, we need to do in interventional platinum chemotherapy. But in advanced stages, the person may have to remove the bladder itself as a treatment, but it affects a lot because the patient has to carry a urine bag all the time for the same. And also, we can see in cases of bladder cancers, the most of the time the patient comes with hematuria, chills, and in some cases persistent urge for urination. And in those cases, the surgery becomes the only regimen for treatment. So for precision therapy, we can look for gene editing techniques like CRISPR, where we can solve common mutation problems in the TERT promoter, FGFR3 and PIK3CA gene segments, which are mostly present in the mutations in the current studies. And also, we can do like less invasive treatments like periodic urine test for finding if any presence of any cells or any elevated white blood cells, or we can just test for PSA specific antigens for any risks related to prostate cancers as well. So we really did very less discussion on this, but we think that it is a much more viable topic, and this the cancer types really needs much more attention, because India is very recurrent in bladder and prostate cancers in recent times. So that's all I want to say. Thank you all, and I want to thank my team for Ms. Khalida and Amar Aziz as well. If you guys have any pointers, you can present it. Thank you.
Thank you, Sandeepan. I think this is one of the successful break rooms that we had, where you talked among each other. This is, I can tell you, a big barrier.
Thank you, Dr. Aziz.
Let's hear from you. Yes, Aparna, you would be the next one.
Yes, sir. Good afternoon, sir.
So, Aparna, you have another 5 minutes, then we'll conclude the session and then we'll have the lunch break. And 2 p.m. we'll reconvene for the foreign faculty presentations.
Okay.
All right. Time starts
So
now.
good afternoon all. My name is Aparna Soni and I'm a master's final year student pursuing biotechnology. I'm from Institute for Excellence in Higher Education, Bhopal. Okay. Unlike you all, I'm not a doctor. I'm just a master's student. So my therapy and all my conclusion is just a smaller one, unlike you all. Yeah. So I would like to talk upon personalized medicine therapy. So my patient is having lung cancer, okay, carcinoma. He's a patient. He came with coughing, cold, blood cough, fatigue, and shortness of breath. He's 56-year-old and an excess smoker, a chain smoker per se. So to take my procedure ahead, I'll take the sample of the patient, pathologically assess his what can I say? We'll go through the experiments, pathological assess them, and we can see that the sample of the patient
Aparna, you're there? We lost you. Aparna, you're not audible if you are there. I think you're there, but
Yeah, so sorry, sir. So yes, we can see that from the patient's sample cell tissue culture, we can see the patient's lung carcinoma. The carcinoma, we can passage it and we can apply it for multi-omics approach as in my approach. Like we can take the personalized therapy to our targeted cell therapy, where we can process the biomarker inside the cell and we can approach like with a nano carrier. We can use a nano carrier as a targeted approach. And I'm so sorry, I'm blabbing. Thank you, sir. I'm unable to speak.
that's fine, Aparna. Good try. I think this is also important that you are able to put your thoughts into words. So, but all of you are doing it on the spot, extempore. So that's also commendable. All of you spoke well, Ilma, Farha, Homa, Padmavati, Sandeepan, and Aparna. Anybody else would like to do it before we close the session?
Sir, may I?
Who's that? Juhi. Okay. Juhi, yes, we'll take you as the last presenter. And please introduce yourself and your.
Yes, sir. Sir, I'm Dr. Juhi Gupta. I'm a dentist by profession and I'm an assistant professor in AMU Aligarh Dental College. And here we come across many oral cancer patients. So I would like to talk about it. And I think we need to go for a multimodal treatment approach for these patients rather than going for the traditional treatment therapy that we are actually doing for such patients. Because if you see the data, at least 1/3 of the patient, no matter what treatment you give, whether surgical treatment or give a combination therapy or surgical or chemotherapy, therapy, there is a relapses. So we should know first, like what are the after giving the treatment, we keep the patient on follow-up. We do the chemo staging before the treatment, after treatment, during the follow-up. But there are chances that some patients they do not come for their follow-ups. And as a clinician, we also do not emphasize every patient, or we do not follow up every patient so rigorously. Like which patient we need to focus more, or we need to do the follow-up very rigorously. So if before doing the treatment, if we do some kind of analysis on these patients, regarding the on the genetics, like the what are the risk factors, like which patient are have more chances to develop cancer later on after the treatment. If we can do that, so there are chances that we can do the treatment for such patient more well, and we can also give the patient a rigorous follow for such patients. There are many ways in which we can do the uh, we can do the treatment. We can do the follow for such patients. Like if before the treatment we have started, we have to look for those profiling. We have to do the genomic profiling. We have to set those targets, like where the mutations take place, like the that the oral cancer, the P53 mutation, the loss of P53 suppressor genes, that is the main cause of for such cancers. And if we look for specifically for some kind of variation in the chromosomes, like 11q13, we have to look for that. Suppose we are going to start the treatment for the patient, if we do the this genomic mapping of these patients, and if we find these targets molecule in those patients, then we can fix our we can make the treatment plan for these patients more precisely compared to other patients. So chances, if we know that the chances of relapse is more in such patients, we have to do the treatment, targeted treatment for such, rather than giving a generalized uh, treatment for such patients or the traditional treatment that we give. Okay. So we think we need for oral cancer, surely we need some kind of this multimonic approach rather than going for traditional approach. Traditional approach is surely it is lagging somewhere, and there are some lacunae that need to be filled. There are many RNA sequencing takes place as compared to normal mucosa that we can target. There are even human papilloma virus in effect is there. If the oral cancer patient also having human papillomavirus infection, that their response changes for the same therapy for the patient who doesn't have those infections. So for this is my take on this. And yeah, surely we should, if we will be, and then I'm also doing some study on use of some nanoparticles, use of nanocurcumin for treatment of these lesions or the lesions that are still in a state to convert from a pre-malignant lesion to oral cancer patients. So that is my take on this topic.
Thank you, Dr. Jui. I think we need to connect and collaborate. We're on the same campus.
Sorry,
Thank
sir?
you. Thank you for your inputs, and we need to connect and collaborate.
Okay, yes, sir. Yeah, that we need collaboration. It is alone.
We are on the same campus, so we can meet sometime and discuss it further.
Sure. Sir, may I have your contact number, sir?
Definitely, we'll provide all the contact numbers and the details soon.
Okay, sir.
Okay. So, Swati, would you please?
Hello? हाँ नहीं मैंने आपको प्रेजेंटेशन दे दिया आपको चल रहा था तो ज्ञान कोर्स कर दिया मैंने उसमें प्रेजेंटेशन हाँ बताइए
Hello. That's great. Thank you. Sure. It says I'm not allowed to share my screen. Mm-mm. Thank you very much, Swati. I'm having trouble seeing my screen as full screen for some reason. So when I did it yesterday, it worked fine. Okay, all right. Let me try it. Yeah, I'll do that. So are you able to see now the full screen or not? You can. Okay, perfect. Let me just go to the. Okay. Can you see my pointer? Can someone confirm they can see my pointer? Okay, perfect. Okay, so yesterday we were discussing how cancer happens, and there were some very interesting and intelligent questions asked. I would request if you have any such questions for today's sessions, please feel free to type them in, and either I can have a look on them while I'm presenting, or we can have a discussion after 40 odd minutes, 45 minutes to discuss those questions. So please do feel free to ask any questions. No question is silly. Okay, so we were discussing yesterday about this compound called benzopyrene, and we had started going into some details of this compound, which is found in cigarette smoke, among other things. And you can see it is not the only carcinogen, cancer-causing compound found in the cigarette smoke, but there are other carcinogens which are also present. I'm sure many of you are aware of that. So what happens in cases, I'm sorry, I've not yet introduced you the terms tumor suppressor genes. I'm only calling them good genes. There is a reason for that. And bad genes, which are, I'm sure many of you, especially those who are faculty members, definitely know I'm talking about the oncogenes. So what happens with the patients who have lung cancer? Their biopsies are taken out, as you know, to define what sort of mutations do they have in their cancer. And it turns out over 50% of the time, the cancers, the lung cancer in smokers have mutations. What sort of mutations? Mutations that inactivate a good gene. Having a good gene function is always good. It is something that prevents cancer from happening in the first place. If you have inactivating mutations, it simply means the gene is not able to do its function as it was required for it. So this could lead to cancer as well. So when samples are taken out by surgeons in the operation theaters, part of the biopsy goes for immunohistochemistry analysis. Is that what was discussed, Dr. Aziz, in the previous lecture? Pathology? Just now, Dr. Aziz? Okay, that's fine. Sorry. Is that what was discussed, Swati, in the just lecture, previous lecture? Okay, so the students are aware of that. Okay. So what happens is part of that tissue goes to a pathology lab, and what will happen is, of course, in many countries and as well part of specific projects, the specific genes may be sequenced. And then what was found that in patients of lung cancer, as you can see, this is over 60%, but at least half of them will have mutations in a TP53 gene, meaning the TP53 gene is not able to carry out its function properly. And you would wonder what kind of mutations would you find in TP53? And this is not the only good gene. There are plenty of good genes, tumor suppressor genes, which may be relevant to all tumor types, or there may be some which are specific to specific tumors. So what happens, we need to see when benzaparin is incubated with the DNA. Scientists have done these experiments and see what changes occur in the DNA as a result of treatment with this carcinogen, which is found in cigarette smoke. And lo and behold, have a look here. This is benzaparin. In itself, it is not really a big carcinogen, but thanks to our liver, it has become a more carcinogen. And what it does in doing so, it actually incorporates this part onto the DNA, making an adduct. So what you are seeing here is that this benzapirin is converting, and what you are seeing changes predominantly. This is the addition of hydroxyl group. I'm not sure if there is any pharmacologist on this call. If there is any, would they would understand what is happening here. So the liver, seeing anything coming into the body, recognizing it is not a biomolecule, it's not glucose. Liver knows what to do with glucose. It's not a protein to be broken down into amino acids. This time it sees it as a foreign and tries to make it more water soluble. You can see this molecule benzapirin is not as water soluble as these ones are by incorporation of hydroxyl groups. Why water soluble? Because then liver can get rid of this through kidneys by making them more hydrophilic. They're more water soluble. But in doing so, it creates products which will actually hook up with the DNA, causing these big bulges in the DNA. So this molecule here, which is formed, this is an adduct. And this adduct will cause a big bulge in the DNA, triggering the repair system. Now, as I mentioned on the previous slide, one of the part of the repair system is those good genes, tumor suppressor genes. And you can see the mutations happening in patients are in the very gene that protects us. So what do you think will happen? In this case, and this is not a reaction happening on just one single guanine. There are millions of guanine. Many of them will have this adduct formation due to benzopyrene exposure. And your guess is as good as mine. What is going to happen here is that these adducts will not be removed because the TP53 function is gone in majority of these tumors, right? So what will happen? Something which was not to start with very carcinogenic has been made more carcinogenic as a result of formation of these bulky adducts. And there is a specific repair pathway in a cell to remove these bulky adducts. One of them is a nucleotide excision repair pathway. So do you think making these bulky adducts is good for cells? No, they're not. At least in this context, this is a normal person who sadly happens to smoke. And as a result, what is happening? Changes are occurring in the DNA. But as long as they have their DNA repair system intact, part of which is, of course, TP53 machinery, they will not see this change in the DNA manifesting itself into a cancer. So they still have ongoing changes, but it will not cause cancer to start with. Over time, what you might see that these changes might actually occur in the very gene that protects us against cancer. And if that happens and the cell cannot correct it, this might actually manifest itself into cancer. Does that make sense? Okay, so one of these products shown on your screen is called BPDEG product. This is an adduct. And what has been found is that in DNA, the formation of this adduct can lead to specific mutations. And there are specific hallmarks of these mutations. What are these mutations going to cause? They can change the nature of a base pair from being a GC to an AT, a base pair. We discussed yesterday these mutations which change purine into pyrimidines and vice versa. We call them transversions. But this is not the only thing happening. What is also happening as a result of exposure to benzopyrene, but also as a result of oxidative stress, we are generating oxidation of some of these nucleotides. So have a look at another. So this is a guanine residue, and you can see benzopyrene product. The product of this reaction, metabolic reaction, has become part of guanine. Where is this guanine? Within the DNA, right? So what is happening? I know there are some questions. I will take them right after this call, Hafsa. So what is happening also is, have a look here. This is a bulky adduct. This guanine has been hooked up with this product of benzopyrene. But also there are subtle small changes happening within the DNA as a result of oxidative stress. One of these changes is shown on your screen. Again, this is guanine, and what has happened here is that there is an addition of this oxygen molecule. We call it 8-oxo-DG. And this is caused by oxidative stress. And when do you have oxidative stress more? In conditions which make you, for example, benzopyrene exposure through cigarette smoke. I have been unwell with the flu for the past 7 or 8 days. And what do you think is happening in the body? It is accumulating lots of oxidative damage. Do I have to worry about it? Perhaps not, because as long as you have good repair system to take care of defects like this one here, or defects like these ones here, these are subtle chemical changes which can actually alter the structure of DNA. That is what we call mutations. If you don't have them, you don't have to worry about getting cancer. We discussed yesterday, there were 3 changes I was mentioning to you. One of them was oxidative damage to DNA, the other one was purine hydrolysis, and the third one was deamination. We will cover them in more detail. But I did point out that I wonder if we don't have any of these happening in the cell, these changes to DNA, and these are spontaneous changes, by the way. They are happening, many of these changes, like deamination, you would see is happening all the time. These are, of course, caused by carcinogens, but in our body, due to oxidative stress, thanks to our metabolism using oxygen to burn out glucose molecules to produce ATP, this is something we cannot avoid. What we can avoid is this part. We can avoid not being exposed to benzopyrene and a lot many other carcinogens. So I do hope that makes sense. And what is happening here? This is BPDE being generated. This is 8-oxo-DG being generated. And both of these things actually can lead to mutations. What sort of mutations have been found to occur? You can see here, this can change, as I mentioned, a GC base pair into an AT base pair. You would wonder, how is this happening? Why the base pair is changing? And we will discuss this. Have a look at this slide. This is an IHC slide, immunohistochemistry slide. So the previous lecture was based on this. I'll give you, I realize some of you were not there in that lecture, including myself. But what happens here, the tissue is taken out. This is a mouse, by the way, mouse tissue, large intestine. Same thing can happen in human. You can take out the tissue, preserve the tissue in a wax, like paraffin, and then stain it with antibodies. It is one of the most frequently used techniques in biological sciences, especially cancer research. And you can see here, this mouse, A is a control group mouse from a control group, while in B, this mouse was fed something which leads to the cancer being formed in the colon, in the large intestine. If you're interested in this paper, please go and have a look at this PubMed ID. This is the ID of this paper in PubMed. You will find that. So you can see here, this is how normal colonic epithelial expression of 8-oxo-DG looks like. You can see here and there is slight 8-oxo-DG. These are the cells within these crypts of intestine. You don't see substantial amount of 8-oxo-DG in this. But in group B, where the mouse was deliberately treated with a carcinogen in order to make it a model to study cancer, you can see here, this stains very beautifully with 8-oxo-DG, and you can appreciate all these epithelial cells are expressing very high level of 8-oxo-DG as detected by this specific antibody. What does this tell you? That the compound used in this, of course, is causing the DNA to have more changes, which is converting the guanine in the DNA into 8-oxo-DG. And that is setting the stage for cancer. So if you keep exposing these mouse, please have a look at this paper, for a certain number of weeks, eventually many of them will develop cancer. Some of these things we may be having through our diet, unknowingly, we don't know, but many of them we know through our lifestyle that we are being exposed to agents which can cause this increase in 8-oxo-DG. I'm not saying, like I was alluding before, I'm not saying that if you are not exposed to these agents, you will not have 8-oxo-DG, but that will be very little. And system is pretty capable, the repair system is pretty capable of taking care of this as long as the repair system is intact. But the moment you overwhelm the system, so now you have a lot of 8-oxo-DG to remove, and consider a situation where these mutations, 8-oxo-DG causing mutations, are happening in good genes, there is nothing to save us from developing diseases like cancer. I hope that makes sense. So I'm going to change, I'll tell you something about these free oxyradicals. So these changes you're seeing here on this slide, benzopyrene, and you can see these adduct formation. This is happening due to cigarette smoke. This can also happen. The 8-oxo-DG can also be found in the DNA as a result of not only cigarette smoke, but also oxidative stress and oxidative damage can lead to this, including benzopyrene and the compound. I'm not for remembering the name, but this compound, which was given to mice, also causes 8-oxo-DG in the DNA. Now, these are the, shall I say, bedrock, these changes in the DNA, these chemical changes, which are making a platform for the cancer to happen. These oxidative stresses are important. But do you know how many diseases, cancer, are treated in clinic? You can give patients the treatment that will cause reactive oxidation species to be formed. What am I talking about? I'm talking about drugs like DNA damaging chemotherapeutic drugs, platinum molecules, platinum based drugs, cisplatin, and so on. What do? They damage the DNA. Do you want the DNA of a cancer cells to be allowed to divide the whole genome? No. So what happens with these drugs? They overwhelm cancer cells by free oxyradicals, which will damage their DNA beyond a point of repair, and these cancer cells will die. You must have heard of the treatments called radiation therapy, and the way radiation therapy kills tumors is also uh, through the generation of free oxyradicals, because that is what it does. High energy causes free oxyradicals to be generated, and that destroys the DNA. So if you see chemotherapy and radiotherapy, in a sense, both therapies are working through a common pathway, and that is through the generation of these reactive oxygen species causing oxidative damage. So if you see here, these mutations, these changes in the DNA rather, these are happening, they are causing normal cells to go on a cancer pathway to become cancer, right? But you don't want these repair pathways, you don't want these um, oxidative damage which is happening in the DNA. Uh, you don't want it in order to get cancer, right? But if someone has cancer, you want to overwhelm these cells with this oxidative damage. Why? Because you want to kill those cells. So how do you overwhelm these cells? You do that by enhancing the level of reactive oxygen species in a cell, or ROS. And how would you do that? Either by chemotherapy, most of the time, by radiation. Both of them will lead to oxidative damage, which will degrade proteins, DNA, to a point where the cell will not be able to repair itself. So the same oxidative damage is bad for a normal individual, which, because this has the potential to cause cancer. If someone is being treated with a therapy to kill their cancer, then oxidative damage is what is needed. Giving them drugs like antioxidants might actually be harming those patients. I hope that is clear. So you can see here what is happening. These papers came in the last few years, and you can see here, this is overwhelming evidence. One paper, this is, I think, Science Translational Medicine. The reference is here. You can have a look at this paper and have a look. Antioxidants accelerate lung cancer progression in mice. Antioxidants can increase melanoma metastasis in mice. So both of these different papers are telling the same story. If you already have cancer in a mouse model, you of lung cancer or melanomas, you can see the effect of antioxidants. They are not good in this context, because here they are making cancer worse. I think one of those papers, I can't remember on top of my head which one, used vitamin E as an antioxidant. Are we seeing more research coming into this area? Not particularly. Why not? my personal view is uh, that it is what is happening is this is a multi-billion dollar industry buying antioxidants. You come to this country, in fact, in India, you can see uh, anytime you go to a doctor, a drug is given and they give you a stash of some sort of antioxidants, right? Do you need them, antioxidants? And this could make actually case worse for patients who have cancer. So you can see here, this is an alarm for those um, doctors or scientists who think that antioxidants may be good for cancer. I was in a cancer meeting last year across Atlantic, and there was clear evidence presented that from an epidemiological study that patients who were prostate cancer patients who had vitamin E supplement actually have progression of their prostate cancer substantially statistically faster than those who did not have vitamin E supplement. So I think it was vitamin E or D, I can't remember, but both of them actually in many ways may have antioxidant responses. So um, I will stop for 5 minutes to take questions. We have plenty of time. There are some questions, and I think it will make the discussion healthier. Swati, is that okay? Okay, let me just have a look here. Um, so the question I have is Hafsa is asking, Walaikum Salam, Hafsa. With respect to BPDE formation, what molecular factors govern its preferential binding to guanine bases rather than other nucleotides? Hafsa, that is an interesting observation you've made. So this is just one of the examples. There are preferences in chemical systems. I can give you some examples later on, but it seems there is an affinity, higher affinity for guanine to be chemically modified by BPDE, and that could be a preferential effect. The exact molecular factors, I will not be able to pinpoint what they are, but this is perhaps just an example. Others may be modified as well with other chemicals on other specific nucleotides, for example. But it seems the benzopyrene prefers to make adducts through those metabolic changes on guanine residues. Then second question again is in the slide I see higher OXO-DG levels in tumor colonic cells. Does this indicate that the oxidative DNA damage is an early event? Absolutely. So these oxidative DNA damage are indeed earlier events. You can open those mice, sequence the DNA, and may still not find any changes to DNA. So there may not be any underlying mutations at this start of these mice being put on those drugs. But if you leave them for substantially longer time, there may have been few weeks, 16 weeks, I can't remember, then it is possible the cells are dividing. Mammalian cells, most of these epithelial cells will divide once a day, sometimes, usually. And what will happen as they divide, they will accumulate. They will change these damage into a mutation. So DNA damage is a different thing, and mutation is a different thing. But DNA damage is indeed, in this case, needed in order to cause mutation. So 8-oxo-DG is not a mutation, it is a damage. But if it is not repaired, it can lead to a mutation, sorry. Early event, you're asking, Hafsa, oxidative DNA damage is an early event in tumor development or is a consequence of tumor progression? I would say the first one. Khadija is asking, so antioxidant-rich food should be avoided by cancer patients. I will not say that. And the reason is pretty obvious. Cancer patients are frail. They need good diet. The amount of antioxidants they will get from these will not be very high to have any effect, any negative effects on cancer treatment. A patient being treated, they will not have these effects because the sheer amount is very little. I can tell you there are antioxidants, you will know, found in green tea. EGCG is one of them. Uh, there are capsules being sold. There are capsules of curcumin being sold in places. And what happens? Sorry about that. So what happens is we calculated that in order to make one capsule of EGCG or green tea antioxidant, you will have to, if you don't want to take capsule, you will have to drink 50 liter of green tea at least. That is impossible. But if someone is taking those capsules, might actually be making their disease worse. Uh, I visited some time ago India, and one of my cousins took me to see one of his very good friends. I was obliged. He's very senior to me, so in age, I went, and they were treating his head and neck cancer thanks to good cousin Chinese. So he developed this head and neck cancer, and perhaps the poor thing died by now. And what happened is they took him to some abracadabra. I'm not sure what it was, herbal clinic, and they gave him a box, and it says curcumin. And I asked him, these must be yellow tablets, and he said, yeah, 5,000 rupees and two of these boxes. Okay, is that what you're having? No, we started treatment and then we stopped because word of mouth, anyone who goes to this person gets treated. Uh, People don't understand cancer therapies don't work like that. And as a result, what happened? You can see this curcumin and other things. They are potential. They have lots of antioxidants, perhaps made the disease worse because that was like big capsules being given to that person. Imagine if this was, you know, in this country, UK or US, people are taking antioxidants left, right, center all the time. Do we need them? What we need is present in our diet. So at least have proper diet to make sure you have everything instead of just relying on enhancing the level of some things which may actually not be good. So as I'm speaking, I may have cells which have mutations, which have changes, but they may not still become cancer because there is a good system to repair those changes. But if I overwhelm my body and have lots of cancer-causing chemicals, what will happen eventually? Cancer. So I hope that answers all the questions. And let's start the session again where we stopped. So you can see from these studies that these papers published in top journals are actually not a good news for people who rely a lot on having antioxidants. I'm talking about general population. They must be taken under. The problem, unfortunately, is, and no offense to doctors on this call, um, we don't know if there are any benefits, but these research suggests there are definitely harm which can be caused by antioxidants if taken artificially, meaning if they are not coming from natural sources like fruits or vegetables, they can actually cause harm. Hmm. So oxidative damage aside, we have this event taking place in the body all the time. We call it purine hydrolysis. And since I started this talk half an hour ago, I guess I probably had billions of these events taking place in trillions of my cells, where the purine is removed from the ribose sugar. It is happening spontaneously in the environment where the DNA is inside the nucleus. There is very little I can do about it. And I don't have to worry about it because hopefully I have a good, and all of us have very good repair system, which will make sure that this DNA, where this is maybe part of a gene or any other area which is gene deficient, this will be seen as a damage and this will be corrected. So just like the adduct formation, BPDE or 8-oxo-DG formation, this should also trigger DNA damage response, seeing this as being damage. And we call these AP sites. So first thing, oxidative damage happening to the DNA. Um, this is purine hydrolysis. And what you can see in the next slide is deamination. There is very little we can do about it. And what is deamination? You can see there are plenty of cytosines in the DNA, and they have this amino group. If you have a look at the structure of cytosine, you'll find that. So what happens here is this can be deaminated. And as again with purine hydrolysis I showed you on the previous slide, this event is taking place spontaneously, this reaction. So what happens, deamination, as the name points out, there is a removal of this amino group. And you will be surprised, some of you, that this this removal of amino group converts cytosine into uracil. Now there's a problem. Uracil can base pair with adenine. That's again not a good news because it will cause mutations. It will change a GC base pair because this is cytosine. If it is part of DNA, on the opposite side of the different strand, there will be a G. So this was GC. But if it is converted into uracil, this can pair up with adenine in subsequent cycles of replication. And that can convert a GC base pair into an AT base pair. That's not a good news. It can cause mutations if it is not corrected. By the way, is there anyone on this call who would tell me what is the target of chemotherapy and radiation therapy? They both generate reactive oxygen species. Maybe you can type your answer if you want to. And we can revisit this in 10 minutes when we conclude this first lecture for today. What is the target of chemotherapy? You may not find it in the books, but something to think about. Chemotherapy, is there a drug target for chemotherapy and radiotherapy? So, is a cytosine the only one undergoing these deamination events? No, not really. There are others like 5-methyl cytosine, which also undergoes deamination, converting it into a thymine. And again, you can see the cytosine, which 5-methyl cytosine, which was paired with GC, as a result of deamination, if not corrected. So if this is not corrected by the DNA repair machinery, what can happen? This will become thymine, and thymine can base pair with adenine, as you know. So again, in both of these scenarios, these deamination can lead to a GC base pair converting into an AT base pair. And that will be classed as a mutation. So this one and this one, both of them. Now, I would want you must be wondering how these changes are happening. You mentioned, so I mentioned cigarette smoke changing the sequence of DNA. How is it happening? I discussed with you these. Okay. Before I'll take more questions. Sorry about that. So have a look at this slide. There are mutations happening as a result of exposure to cigarette smoke, among other things. And can you tell me, I'm seeing some strange changes on my screen. Can you still hear and see the slide, Swati? Okay, thank you. Sorry about that. I think I'm new to Google Meet. I usually use Teams, so sorry about that. Look what happens here. There is a patient who is a smoker, and as a result of this, what you would expect to see in them is the formation of 8-oxo-DG. So this highlighted G is now something which has become had either 8-oxo-DG or a BPDE. In both cases, what is this is G, which has changed due to oxidative stress. Something similar you would learn can also happen with apurinic sites, meaning where the purine has been taken off the pentose sugar in the DNA. And look what happens here. Uh, DNA replicates, and DNA replication is semi-conservative. So each strand of the DNA, this is textbook biology. Um, you can see here, this is strand and this is strand. Each one of them uh, can actually become the template for the synthesis of new daughter cells, right? So those daughter cells which harbor this strand, where this strand, which is not damaged at all, serves as a template. What will happen? You see any mutations? Not at all. So in the new strand, in the daughter cells, there will be no changes. So one of the daughter cells, which is a progeny of this cell with a mutation in one of the strands, sorry, with a damage in one of the strands, this cell will be perfectly fine. There will be no changes in it in terms of the DNA structure, sequence of DNA, sorry. And look what happens here. Uh, if this strand, the upper strand here, DNA, which has the mutation, sorry, I'm saying again the mutation word, which has the oxidation of this guanine, if this strand were to serve as a template for the second daughter cell, you can imagine what is happening here is that instead of a G, there is a G, there must be a C. Now the system, the repair system will bring an adenine. So the system usually will bring anytime there is a failure and there is the oxidated G, an adenine will be brought. Should there be an adenine here? No. There are pathways, which we will discuss in about half an hour, which will see this as a defect and they will correct it, hopefully. So what happens here is this daughter cell is not looking good because it has now accumulated a change in the DNA. It should have been G, that in the new strand shown in red, there should have been a C, and that is not the case. So that cell is not looking good. It's not a healthy cell. And if a repair system is not able to see this as a mismatch, this is a definite mismatch, you can see there should have been a C here, then this mismatch can lead to first cancer cell being formed. So this is something that can happen as a result of failing to correct the DNA damage repair. And have a look what is happening here. This is the last slide. And then I'll take more questions and we can have a break of 10 minutes. So what is happening here? 8-oxo-DG, you can see this is formed in a double-stranded DNA. There was a cytosine. And you can see here, when the DNA is replicating, each strand is serving as a template. One strand was fine, as one daughter cell, one strand was okay. And in that cell where this strand served as a template, there are no changes. In the other one, of course, we have now a mismatch which needs to be taken care of. What if this is not taken care of? You can see this adenine comes in here, and if this is allowed to replicate this cell, this monster cell is allowed to replicate, what will happen in the subsequent cycle at this location in the DNA? The base pair, which was originally GC, has now actually become an AT base pair, which is a classical definition of mutation, changes in the sequence of DNA. What about these AP sites? So you have these purines gone, for example, this G is not there anymore. This is happening, as I mentioned, spontaneously. What is happening here? Again, the AP site, if this strand, the upper strand, were to serve as a template in through DNA replication and the formation of new daughter cells, if this DNA strand were to serve as a template, you can see what will happen here is that there was a G, which is now gone. And what will system bring here is an adenine. And if this DNA with this mismatch is allowed to become is allowed to proceed without being repaired. This will change in the next cycle of DNA replication a base pair which was originally GC into becoming into an AT base pair. Again, that's a classical definition of mutation. So with this, I will stop and I'll see what questions are there. And Swati, if you like, you can switch on the speakers as well. So these students and colleagues can ask me questions directly. In the meantime, while you prepare yourself, I will take questions which I see in the chat box. So yeah, I think I've all answered some of these questions already. Antioxidant-rich food should be avoided by cancer patients? Definitely not. What should be avoided is super high, ultra high doses of antioxidants given as tablets. That is my personal view. Padmavati, you're absolutely right. Chemotherapy and radiotherapy cause the target is the DNA. So chemotherapy does have a target, which is a DNA replication. Afi Afzal saying nitrogenous basis of the DNA. Yes, that's correct. Basically, it's a DNA. The target is DNA. But Mawati is saying forms DNA adducts with nucleotides. You're absolutely right, such as cisplatin will make DNA adducts. So those DNA adducts, I just gave you an example, which can make a person sick by leading them to a cancer path. Those DNA adducts are needed in order to kill cancer cells. Fatima Khan is saying chemotherapy interferes. Absolutely right. Uh, Radiation therapy causes direct DNA damage. That's correct. Direct simply means they will lead to generation of reactive oxygen species, which will cause oxidative stress burden on cancer cells and causing their DNA to get damaged permanently. So Nilofer is asking, why is deamination considered an important source of a spontaneous mutation? Your question has its own answer, Nilofer, because it is spontaneous and it is super frequent. It is happening at enormous levels. Since I started this lecture 45 minutes ago, millions of these changes have happened. There are chemical reactions. You can see within the environment, aqueous environment of the DNA. This is one of the changes happening. And I did not point to you, and perhaps we have time for this, Swati. If I go back to this slide, we all studies right from the high school. The thymine is present in DNA and uracil is present in RNA. We all know that. Looking at the slide, can you tell me a reason? You can activate your mic if you want, or you can type this question. Those of you who want to take a break, we will meet in 13 minutes again. I'll stay behind before taking a 5-minute break. But one of the answers why you see uracil in RNA and not in DNA, and in DNA we have thymine. You can tell me the answer now, looking at the slide. So Madiha is asking, in the meantime, Padmavati is saying, thank you, no problem, Padmavati. Madiha is asking, sir, so do antioxidant supplement worsen cancer by protecting cancer cells? I will show you some evidence. I've already shown you some evidence from this very strong Science Translational Medicine Journal papers, which suggest actually this is the case. Antioxidants can make the treatment of lung cancer, the progression of lung cancer worse actually. Because while you're trying to kill those cells by generating oxidative reactive oxygen species, those antioxidants are going to remove them. You can ask yourself, is it good? Is it bad? And you would see cancer is not something that all of us should worry about. I know the cancer cases are on rise, but in the next lecture, which will start soon, you will learn that actually is.
Can I answer your question, Dr. Asim?
Yes, Dr. Aziz, please go on.
We put a 10-pound reward on them.
On what question? On this?
Why DNA has got thymine and RNA has uracil?
This is one of the answers I'm showing them on this screen. So I've given already a clue.
That's nice. Anybody can answer that question? That's a very simple fundamental thing, and the answer is right there. I think the post-lunch
But yeah, Dr. Aziz, yeah, I mean, when they're thinking about this, this is quite alarming. I'm sure, Dr. Aziz, you must have seen in US, you go and walk into a pharmacy and all you see is these big aisles of antioxidants where these are not regulated by drug regulators. You can go and buy as many as you want. Do you really need them? We need to ask some.
Yeah, but still in US they do test it for the toxicity and other things, so they at least don't have the ingredients like excipients which are toxic. So by and large they remain harmless unless you overdose them.
Okay, so these antioxidants, I was telling these papers which came out in Science Translational Medicine, actually antioxidants are bad for cancer.
I think it depends on dosage, that how much of it are you taking, right?
Yeah, so I gave them an example of EGCG found in green tea. What you get in the market, pure EGCG, to have the effect of one capsule, you will have to drink 50 liters of green tea, which nobody does. So you're actually artificially increasing the level of antioxidants in your body. Is it really warranted?
True.
So I think there may not be any questions, Dr. Aziz. Shall we take a 10 minutes break and reconvene?
Yes, absolutely. And we can keep the session live and on. So please,
let's do that.
Question and you can post
if
it.
you have any questions,
or I can tell
type
you
in the questions.
on. So if anybody wants to use a microphone instead of typing, that's fine as well.
At least use this as an opportunity to get trained to ask questions.
Sir?
Yeah?
Sir?
Yes,
Dr.
Juhi.
Juhi here. Yeah. Sir, I learned learned about vitamin E, the role of vitamin E, and the probable role of vitamin E in worsening the lung cancer.
That is the evidence from this paper.
Sir, so is there like I have gone through many articles, but they have told that if we are using it for a very high dose, means when the doses increases more than 400 milligram per day, it seems. Is there anything like that? Like if you give it for a long period, and also the dose matters.
I think what you're absolutely right. You have a point there. So how much you're giving, what I believe is what is needed is in your diet already.
In that,
yeah, I mean, reaching 400 milligram, you said, it would be impossible. Even if you get a truckload of food and eat them, you will not be able to, not in case of vitamin E, it may be called liverol or something like that, because it's hydrophobic. But actually, it is quite alarming with the way we have becoming addicted to using these antioxidants as a supplement. That's what I'm saying. May not be a very good idea, actually. And we all know, cancer is the last manifestation of changes, mutations that happen within the DNA. And there is very little you can do many times about this. And the best drug.
No, sir, actually, I am doing one. Yes, sir, I am doing one clinical, one randomized control trial. I am a dentist by profession, and I am assistant professor in Dental College, AMU. And we get many cases of pre-malignant lesions, like oral lesions that may convert into malignancy later on. So in that, I'm giving vitamin E in a very low dose, like it is 200 milligram.
Okay.
Like that for 2 months, not more than that. Means I ask patient to stick to that regimen for 2 months only. Some people, when they are not, because it is very cheap also, sir. That capsule, 10 capsules, it costs not more than 15 rupees. So if they are not coming for a follow-up, they are themselves buying it and having it. So I have told them many times. This is a problem in Indian scenario, that that I am facing. Even though we are telling them, please, it has to be only for 2 months. Do not go beyond that. So yeah, sir, I
Yeah.
am totally agree. I usually counsel patient to modify your diet. Get the nutrition from your diet. But then, like, ma'am, it's not possible for us. What you're talking about, to have like salads, have cucumber, have tomatoes, like that. So you please
Mhm.
tell me some antioxidant that we can take. It is
Dr.
happening. So now, how to
Dr.
deal
Gupta,
with such patients?
Dr. Gupta, that is a problem here as well, that people just go and buy, because as I said, this industry is not regulated by drug regulator. So you can go and buy anything you like, but you are
Yeah,
doing an
it's
excellent trial.
such a OTC, such a common OTC present. Like you can buy anywhere.
That's
So
right. I think the
no,
trial
at
you're
least
doing is excellent, because
no.
you're seeing these pre-cancer fibrotic lesions probably, and
Mhm.
giving them before cancer actually happens. So
Yeah,
that
that's
might
what you
actually, that's excellent. That's a very smart idea, actually.
And I'm doing the biopsy also at the end of the treatments, just to see if
That's great.
any adverse effect has had taken place. Till now, no patient has developed any. If the patient has stick to the regimen, I'm targeting only those patients.
No, that's
That
good,
is
and especially because it is in the context of pre-cancer, you may be able to extend the window before they develop cancer. And as you rightly said, some of them may not even develop cancer. Wonderful.
So I'm really sorry for the interruption, but there has been some problem.
Can you not hear me very well?
Yeah, but it's echoing.
I think it is maybe echoing because someone has left their, they're not using microphone, headphones maybe. That's why it is echoing.
Okay, I'll check.
Yeah, can you still hear the echo?
Dr. Juhi, can you
Hello.
turn your mic for a second so that I can hear?
Let's
Okay,
see if it is echoing. So testing for the echoing of my voice. I think that's why this was happening. Okay, thank you. Yeah, I would say this is exactly what we need to do. India is such a big country, and even in centers like AMU. Actually, one of my friends graduated from that dental college, the inaugural batch of the dentistry. It's actually a good thing, because with these gutkhas and khanis, you see these patients tobacco consumption. The cases are on the rise, and as I mentioned yesterday, this is the last thing one would worry about. There are reports of people not being able to make rational decisions, depression, cardiovascular effects of these consumption of tobacco, gutkhas, and so on. So this is all. Congratulations to you and your team, Dr. Gupta, what you're doing through this RCT. That's great.
Thank you, sir.
Wonderful. Let's touch base with us. You have my email address. Let me know how this trial goes. Once you have the outcome. Wonderful. Anyone else with any questions, or shall we take 2-3 minutes break if everything is fine with that? Anyone needing a break? Yeah, if you need, we will keep alive. You can ask me questions if you have. So, Dr. Gupta, I met this patient. I don't treat patients, as you know, I'm a scientist. But looking at those reports, they have local invasion, and instead of going and taking the patient to PGI, they preferred giving them tablets of curcumin, hoping that it will treat, because it word of mouth, many people go to this person and they were all cured. Cured, I don't know what cure means, but basically this is quite on the rise in India, which is quite alarming, as I see it.
Yes, sir. That is the problem, actually.
And it's very difficult. You can't really, I have relatives, my first cousins and some families, my uncles and aunties, some of them developed cancer, died of it, or are in the treatment. it's very hard to make them convince what is the best course of action. They don't realize, they don't appreciate. Anyone tells them, oh, this Baba in Ghaziabad, they have a queue, people queuing up. Yes, lots of silly people going and queuing up. They're treating cancer left, right, center, and they go there, pay a hefty amount of money in a hope to get treated, and end up actually getting quite ill and die a very sad death.
True, sir. There are many cases in which I have advised patient like you should go for biopsy and go for the treatment, but they have not gone for the biopsy. They have gone to some Baba and someone, and then they are returning back with full blown stage 4 cancer, which I have seen in stage 1 has gone to stage 4.
Goodness. I hope your trial and those who stick to your adherent treatment will make a lot of difference. Because you can see these fibrotic lesions, right, before it happens, or?
Yeah. But there will be white patches. There will be fibrotic lesions will be there. So we are giving treatment and we are doing now. Like whatever the treatment already going on and what I am introducing in that, I'm comparing.
That's great. I was seeing a report some time ago that they were doing some trial on epidemiological studies of head and neck cancer, actually oral cancer, in people who were going to who drink very hot tea in Iran versus those who never drink any tea. And there was some association I vaguely remember with the hot drinks. So anyway, Dr. Aziz, do we have your permission to crack on if there are no questions? Let's wait for a couple of minutes. Shall we start, Dr. Aziz, Swati? It's being recorded anyway. So yeah, please go on. Okay, that's good. Let's crack on with this then. Okay, so yes, we were talking about these changes which we have very little or no control over. Why we can have these spontaneous changes in the DNA, and how it can lead to permanent accumulation of mutations in the cells. So I think I covered all of this. Okay. And you can see here what is happening here. In both cases, whether this is 8-oxo-DG, which is the oxidative stress causing changes, subtle chemical changes in to the DNA, converting GC base in a double-stranded DNA as a result of mutation, as a result of replication into an AT base pair. And likewise, these apurinic sites, this is one of the most common change in the DNA happening again spontaneously. This can change a GC base pair into an AT base pair. So you can see here, and you can start expecting what will happen to patients who have mutations if you isolate their DNA and sequence it, the DNA base by base, and compare it with the reference DNA sequence from a normal individual. And we won't go into the genomic level, but we will have a look at just one of the genes and have a look what is happening here. This is the data for TP53 mutations in patients who are smokers. Do you see any patterns? Let's start with this one. Have a look what is happening here. You see that there is a higher frequency of Gs converting into Ts. So initially, in normal individuals at specific nucleotide positions within the DNA of this gene, there are Gs. And now, in smokers, they are mostly Ts in those locations. Where is it coming from? It tells you exactly that this is caused by the cigarette smoke, which can convert a GC base pair into an AT base pair. So what was initially G, instead of this GC, now there is an AT. And that is why you can actually get a clue of where this mutation is originating from. And this is just one of the carcinogens. There are thousands of different carcinogens, different degrees of the way they mutate the DNA. It is known. The way they can change the structure of DNA may actually influence the diagnosis. If you're seeing these sequences changes in specific nucleotide positions, they may be a result of specific carcinogens, such as this one being more common in smokers versus those who don't smoke. You can see there is a substantial difference between them. Now, yesterday, somebody was asking me, and I am asked this question a lot of time by family and friends back home in India, also in UK. I have some family here too, about people who never smoke still develop cancer, and that gives them a very silly reason to not stop smoking, thinking everyone gets it. But there are 25% people who do develop lung cancer, sadly having never smoked. But then we develop many other cancers without any known etiology as well. So have a look at this landmark paper which came earlier this year in none other than published in Nature earlier this year. And this is talking about the mutations which happen in lung cancer in never smokers, short for LSINs, as you can see. So this paper talks about many different changes that happen in patients who develop lung cancer, but sadly, they develop it despite having never smoked. And what they did, they had a look at the mutational analysis within this paper. Have a look what is happening here. So they collected samples from 871 individuals, since individuals. So they have lung cancers in non-smokers from 28 geographical locations. Some of them are, you can go and have a look at this paper. If you don't get it, please write to me and I can send you a copy of this paper. This should be freely available to everyone anyway. So the most, they've used few things in this paper. One of them is the AQI or PM 2.5, which is one of the measures for air pollution. And they have countries like Azerbaijan in Eastern Europe, Hong Kong in Far East, as examples of very bad AQI. I'm talking about hundreds. And they collected the data, and you can see here that there are increased levels of PM25, which is one of the components, major components, and the DNA damager within the air pollution that can cause an additional DNA damage. So imagine we have all these processes destroying the DNA. They're making the changes, purine hydrolysis, AP site generation, deamination, and someone is a smoker. And on top of this, if you have air pollution, have a look, by the way, this is an excellent paper in this study, and they show what is happening here. You can see these are the increased levels of so what you're seeing here, sorry, I'm trying to coordinate the screens. So you're seeing these increasing levels of this pollutant, and patients experience mutations in DP53. So that's a good gene which has bad mutations, meaning it's not going to function properly. So this is one of the most extensive studies I've seen, which takes into consideration a lot of cases of LSINs, cancer of lung caused by environmental conditions. It is a very bad news. You can see here these PIK3A mutations, KRAS mutations. So KRAS is one of the drivers. Mutations in KRAS are very common in certain cancers, including lung cancer, and you can see many of them are enriched in a patient. They have other slide in this paper, and they show the mutations which are enriched in Asians, patients who are all as these patients, so they're never smokers or excess smokers at least. And you can see here they compared Asian mutations with North American or Europeans, and you can see here in Asians, these again, the mutations which are most common are the mutations in TP53 or this EGFR, which is a growth factor driver, and we will learn today, not today, next lecture, how does EGFR manifest its effect on once it is mutated on the growth of cells, especially with regards to cancer. So very interesting study to look into, and this is quite alarming. They have, I was looking at Azerbaijan and Hong Kong. The AQI is really bad in those countries, can lead to actually real time. And this is, of course, association studies, but you can see there are other studies which are referred in this study showing that, in fact, there is a lot of harm which is possible through exposure to environment pollutant to human body, and that can lead to this. This will set a perfect stage for cancer. You can see it this way. So how do you identify what is causing cancer? There are lots of compounds people think might cause cancer. Others, people never doubt this will cause cancer. And there is, on next slide, is very simple test, and this is called Ames test. I will discuss this briefly. Sorry, I'm seeing a question while I'm speaking, and Ilma saying, maybe because of AQI 700. Yes, I'm in Delhi NCR. I don't know about Delhi. I used to live in Delhi for some time. Where I'm sitting, the AQI currently is 33. People still complain about London, which is the next city where the AQI is 50. So coming back to the topic again, have a look at this test. And this is a very simple and very smart test. It can help you identify chemicals which are direct carcinogens versus chemicals which are indirect carcinogens. And what this test involves is using the liver extract. And what you do, you take a suspected mutagen, incubate it in a liver extract, and plate on an agar plate. Now, this is not just any agar plate. This plate does not have histidine in it. Bacteria need histidine to grow. So what will happen if they don't have any histidine on these plates? These bacteria will not grow. Which bacteria are these? These are hist-deficient salmonella. They're not going to grow in this extract. So in this test tube, you have three things. One of them is liver extract. The other one is suspected mutagen. And the other thing is the bacteria. In this control tube, you can see there is no mutagen. Nothing grows here, of course. But what you might see that if something has the potential to cause mutations, actually they can remove this defect in the bacteria. There are very subtle defects that make this bacteria not capable of making its own histidine. Just like defects I was showing you, very small defects in previous slides, where a GC, for example, converts into an AT. So you can imagine this as something which has been changed by just one nucleotide. And by doing so, the bacteria, the histidine synthetic synthesis by pathway will be deactivated. By reverting this change, you can activate this pathway. And if that happens, what would you see? You would see those colonies forming on the plate. And that would suggest that this is actually now active. Why liver extract? Because liver extract will allow those chemicals which by themselves are not carcinogens become carcinogens. They can now cause cancer as a result of changes through the metabolic pathways. So if I show you some slides, these are again cartoons. You can see here, this is a background experiment control conditions. You might see some colonies. You always have some contamination. And you can see here establish whether what happens if there was a compound which was a direct mutagen. What will happen is, even without needing the liver extract, you might see a lot of colonies, meaning it has now converted, it has reversed that defect in the histidine biosynthetic pathway. And now a lot of bacteria are actually able to grow and form colonies on that agar plate, which was deficient in histidine. And then there are some examples. Think about these ones. What happens here if this is similar to the background, and you can see this is not a carcinogen. You see more colonies, and compared to the control, of course, this compound is going to be a carcinogen. And if it is going through liver metabolism, what is happening, that this may be the result of this not having the liver extract. It is still a carcinogen, perhaps a weak carcinogen, shall I say, just like benzopyrene. But what happens if you have liver extract as well, then it becomes even a stronger carcinogen. So that test is a reflection. It is directly proportional to the extent, the potential of any chemical to cause mutations. So it does not directly detect carcinogens, but we know cancer is a manifestation of mutations in the DNA. Then many of these chemicals will be potentially carcinogenic. So that's an excellent test for carcinogens to establish their identity as chemicals with the potential to cause mutations. But how do you test them? The person who tested them first and is credited with developing the first model of cancer is Yamagiwa, a Japanese scientist. And what he did is he took the rabbit and painted it with tar, crude coal tar, the inner ear. And in doing so, they were able to see some changes in the cell, not physiology, cell behavior, and identified later on that actually it was making cancer developing uncontrollably, the cells within the inner ear. And this is just a crude coal tar, must have lots of multiple, numerous cancer-causing chemicals. So he was the first one to establish this. I was showing you a mouse colon model before, where I showed the accumulation of 8-oxo-DG. That was part of other model. You can use those chemicals to induce tumors. Now you will ask me, why do you do such cruel experiments? Especially in UK, it is a big problem with people sometimes coming and vandalizing labs. They don't want any experiments to be done on animals. Little do they realize that the vaccines they all have been vaccinated for, saving them from infectious diseases, actually come from animal experimentation. So there is a big debate. Should we not use animals for experiments? Should we use animal for experiments? There are ways where we can avoid use of animals in experiments. These are harsh, cruel experiments, but in UK, and I'm sure similar things have regulations exist in India, where you can't really give harsh treatment to animals or increase their tumor load beyond certain point. In UK, this is 10% of the body weight of a mouse. So imagine a mouse being a 25-gram, 3 or 4 week old biopsy mouse, 25 grams. You can establish a tumor which should not go beyond 2.5 grams when given through subcut, which is under the skin. So what we have learned are different dots today, that there are chemicals which can cause damage. Do we not get damage in the DNA? We do get damage to the DNA anyway, all the time happening spontaneously. What people are doing by exposing themselves to cigarette smoke, for example, is enhancing those damage, making them perhaps beyond repair. And that will be the first step. A cell will divide, and this cell will divide, and a daughter cell will harbor a mutation. It will not be a mismatch. it will be a proper mutation where at a particular locus within the DNA, the nucleotide has changed. Would that cell become cancer? Definitely, the answer would be, in my view, no. You need those mutations happening to that daughter cell again and again, new mutations, which will make it more fitter, or shall I say, more rebellious, which will not follow the principles of normal physiology and anatomy. And in doing so, the cell will become cancer. So let's go and talk about this. You can see this slide. I'm not talking about animal migration. Why am I showing this? Have a look. This migration taking place in Europe. These birds go as far as taking a flight from London to New Delhi airport, over 7,000 kilometers, and they carry out this journey. No doubt, you can see some people observing this. Uh, this requires lots of adaptations, lots of endurance exercises to be able to carry out these journeys. And we know these animals are adapted. Not every bird will carry out these migrations over mountains. You can see these birds flying over clouds and mountains. They go as far as Himalayas, or on top of Himalaya, which is I think 8 kilometers or so. They need to be specially equipped. And this is why am I showing this? Because this is something similar happening in cancer cells as well. They need to be extremely fit in order to survive many journeys, to allow them to go from one part of the body to other. Have a look at this slide here. These are some of the journeys taken by cancer cells. So what you're seeing here is from a mouse. You can see the liver is completely patched up with melanomas. The cell from the skin's migrating, melanocytes migrating into a lung. You think this might be a simple journey, must have taken a lot of endurance for these cells to have a lot of resilience. In some ways, I have respect for cancer cells, really, with the damage nature gives them, and they still survive that journey, becoming more resilient. With the example I showed it to you, they are much more fitter than many other cells, and I'll show you some weaknesses they have as well in the tutorial, hopefully today. You can also see this similar journey taken by these cells. You can see melanocytes sitting inside the body. As a result of migration, you only find melanocytes on the skin. You can see some spindles, the blood forming, the milk making cells sitting in brains. And you can see also these colorectal cancer cells sitting in a liver as a result of taking this long journey, as I was mentioning to you. So for organism, of course, this is not a good news because it kills those patients. Sadly, all of them would have died of their disease. These are probably from autopsy samples, definitely these ones as well. So they need to be extremely fit. And that's a good news for us because in order to become trained to be at that level, it takes a very long time. In fact, in many of them, many of us never develop cancer. So how many roughly develop cancer? I think every 3 individuals in 5, so 60% develop cancer, and rough estimates are that 40% are killed by cancer. So why this is needed? They need to be highly resilient to carry out this journey into distant organs. These cells were never meant to be here, but they are actually surviving here and killing the patient. So cancer cells, actually, they have a lot of barriers to cross. They need to be extremely fit in order to migrate from their primary organ to distant organs. And this migration is, you can show here, this is shown as these athletic games, or a swimmer which is trying to swim away from a shark, I believe. They need to be extremely fit. In our body, the shark will be none other than our immune system, which will try and stop them in their tracks. So they need to defy a lot of odds in order to carry out this journey and come to this stage. So when I'm saying defy odds and migrate, metastasize, what am I talking about? I'm talking about what is shown on your next screen here. You can see what these cells are capable of. As epithelial cells, as I discussed yesterday, they form surfaces of different ducts. They form surfaces of our skin, uh, different organ surfaces. And you can see here, they're meant to stick together. They have a glue called cadherins, E-cadherins. I'm sure many of you know E-cadherin proteins. And their primary role is to stick the cells together on an epithelia, forming a covering. That's what they do. But these cells are undergoing these changes. Uh, is there any question? I heard someone. Padmavati, do you have any question? Okay, sorry. So what is happening here? They were meant to stick together and form the covering in order to protect the inner surfaces, inner linings of that organ. They're not doing it. And you can see here the normal cells shown in yellow, but these cells which have the primary mutations, these are the cells which have the initial DNA damage. Sadly, the repair system failed. They were not able to see these. And what will happen? They will have certain mutations in them. These mutations will allow them to perhaps outgrow the other cells. So you can see here, these cells, there must have been a single cell shown in blue. They are overgrowing now compared to the other cells. So they're already showing rebellion. They're not really following. They should not be growing as they are growing at the moment. They are starting to grow more. This might require mutations. And trust me, you go and have a look at any cancer. There are hardly any cancers in which there are no mutations. You will always find mutations in cancer cells, uh, provided your sampling is correct. So what happens here? That's fine if they are growing. But what happens over time when they accumulate one mutation after the other? Imagine this cell, the initial cell, which I was showing you, they were not able to correct with this monster face I showed you, an emoji. They were not able to correct the defects that they have. And because they cannot correct, they have first mutation. Chances are that they will have more mutations because they have run out of the repair system. If they have the mutations and they are allowed to grow further, what can also happen is that they can start attracting the blood vessels. In fact, they can start to make their own blood vessels supply, a process we call as angiogenesis. So they're capable of making their own blood supply in order to escape the primary site. In fact, there is a recent paper which came, I don't want to quote a wrong paper, but it shows that if you treat primary tumor, sometimes you can promote metastasis. I'm not saying don't treat primary tumor, but have a look at that paper. I think that paper came in Cancer Cell. I might be able to find a reference if you need. But have a look what is happening. You need these cancer cells to be much more fitter, like those birds, migratory birds I was showing you. They have lots of adaptations, as you know. So what happens next is that they start to invade. They are not meant to melt the connective tissue underlying where they were sitting. They start to melt the connective tissue. Specific enzymes they have you heard of metalloproteases? Many cancers overexpress, they have more metalloproteases. And they're basically melting anything, any connective tissue, any stroma that come in their pathway. What are they trying to gain? They're trying to gain access to the body highways, which are our blood vessels. Because if they can gain access to this, they can go anywhere in the body, anywhere where the blood will take them. Sorry about that. And what is needed in order for them to make these metalloproteases? Epithelial cells don't make it, but these cells gain a property to make those metalloproteases. And do you know why this is happening? Because the genes were already present. They were epigenetically, as Dr. Aziz was telling you yesterday, and Dr. Hibs or Siddiqui will tell you, I think tomorrow or day after, that there are epigenetic changes. So genes, specific genes are switched on in cells, specific genes are switched off. We all know this as being selective gene expression. We know there are pancreatic cells, specific cells in pancreas, which make glucagon, which make insulin. Not every cell can make insulin in our body, can it? No. But the gene for the formation of insulin is present in every cell because the genome is constant. Every cell has the same gene set. It is only that those genes are not expressed. But what these cells are doing, they are expressing those genes, and by doing so, they're able to melt these connective tissue, gain access in the blood. What are they going to encounter in the blood? Lots of cells, from lymphoid cells, lots of immune cells, lymphocytes, which should attack these cells and kill them. And yes, believe me, it happens. So although the first cells might have been formed, pathologists take a look, very dense chromatin, it looks like cancer. It may still be very differentiated, meaning that may not be an advanced cancer. So what happens for cancer to form this, for cancer cells to gain access, for cancers to survive this journey into the blood, and for cancer cells to go and sit? And these are not whole big lumps of tumors, they're single cells moving. It requires mutations after mutations after mutations, which make them fitter. And as a result of this, what can happen? They can gain ability to sit in lungs and grow. They prefer bone a lot of time because then they can hide sometimes from the immune system. But more importantly, bone is very high in nutrition. Almost 80% of human tumors have been reported to metastasize, to take this journey and sit in bones, exactly like those birds I was showing you who are going from cold, harsh weather, winter from Siberia into the summers of Asia, for example, doing this journey. And some of them actually might sit in a brain because in all of them, their own perpetuity will be guaranteed. And once they reach these organs through blood, they need again the capacity. So remember, these cells are changing their not only their behavior, but they're expressing specific genes. How this all happens, we don't know. We know very little about this. You see some papers being published every day, which would show some of these characteristics of cells becoming cancer, carrying out this invasion metastasis cascade. But how it happens in a live human being, we are far from knowing this. And in the end, what happens here is these cells can go and sit in different parts of the body. So that is the entire journey completed. And you can also appreciate these epithelial cells are different. These are more like mesenchymal cells when they're flowing in the blood, and then they regain their ability to sit back in the target organs where they metastasize to. This transition has been dubbed as a epithelial mesenchymal transition. And you can see here what is happening in epithelial mesenchymal transition is that this is the tumor mass, and I'd want you to focus on cancer cells. Shown here, you can see here, these are cells undergoing from epithelium becoming mesenchymal, meaning they're growing legs in order to move out of this organ. Demand for nutrition, right? They move out because of this. They want to proliferate, they want to grow, they want to take hold of other lands. So what they're doing, they're moving out of their primary organ, they're melting the connective tissue, gaining access of the blood vessels, going into the new organs, for example, bones, where they're full of nutrition, and establishing their colonies in distant organs in order to metastasize. You can see it probably as like British Empire leaving very cold, harsh island of United Kingdom, Britain, and settling in many parts of the world at the expense of the local population and enriching themselves from it. This is a perfect example of this. You can see this in that way. But you can see it didn't happen overnight. It requires a lot of changes in the DNA to make them fitter, where they are able to change from being the epithelial cells into being mesenchymal cells, and then from this state, these cells being able to travel and survive into the bloodstream, and then going back into their epithelial form and start forming tumors at the expense of the local population of cells. Can someone say that they can hear me? Yesterday we lost connection and there was no one who. Swati, am I still audible? Okay, thank you very much. So you can see this. Thank you very much, Dr. Aziz. So you can see this is not an ordinary journey. It requires, each step of it requires these cells to become fitter. As someone of my age, I suspect I have some of these cancer cells already, given my parents' cancer, died of it, may have these cells, but I don't want you to get worried, but see what happens. We have a very good system that keeps us safe. They need to have lots of mutations, right mutations, that will not only make them grow at the primary site, but actually be able to metastasize. Each of these events will need new set of mutations. Of course, if you have one mutations, there will be more chances to have subsequent mutations in the same daughter cell as it divides, but that will take time. So the essence, the crux of this happening cancer, it needs to have enormous level of replication events. And that replication event will allow it sometimes to accumulate right set of mutations which are needed for the cancer cell to carry out this entire journey shown on this slide. So where do you think you would see more cancer forming? We have different cell types. Have a look at this slide. Okay, this paper came, I think, Nature. Sorry, the reference is missing. You can see on your screen, on this y-axis, there is a lifetime risk. Lifetime risk is going up for different cancers. And on x-axis, you are seeing the stem cell divisions. Where do you see the most stem cell divisions in FAP, this specific colorectal cancer that Dr. Aziz's lab studies? You can see that we have a very high lifetime risk of developing colon cancer because they are the ones which divide the most. And of course, division takes very, very long time. So if you're dividing, if you're a cell who divides by tendency a lot, then the chances of having this division and accumulating those mutations is much more higher compared to those cells. For example, these pelvis osteosarcomas, head osteosarcoma, you will have to go cities to find cases of these cancers because they don't really divide. And as a result, the lifetime risk of developing such cancers is much more lower. I was showing you that Nature paper which came about the air. Sorry, is there any question, Dr.
Yeah,
Aziz?
I have a question here that always bothered.
Sure. Sorry, I lost you. I think you're mute.
Sorry, what about the pediatric cancers? Cancers in the
Excellent question you have asked. So, do you see any pediatric cancer on this slide? For example, retinoblastoma and so on. So all of these cancers you're seeing, they have very high division or low division. Pediatric cancers are usually exceptions because, for example, in retinoblastoma, the cancer of the retina, back of the retina, you have just one mutation in single gene. Sometimes in retinoblastoma gene, RB gene itself, that's a single mutation that causes that good gene to lose the ability to control a transcription factor, which is E2F. I'm not going to the detail as yet, but I will. What happens there is that they are inborn, they are germline mutations inherited from parents, where there is just single mutation. They don't have any other mutations because the cells have not divided at all. And eye cells, retinal cells, for heaven, don't really divide so much. But those mutations which are found in babies actually can lead to development, abnormal growth of the cells in the back of the retina, and they can actually migrate into the areas near the head, not migrate, but invade into the local areas. So as such, they don't have very high cell divisions in them, but they're still capable, not because of the tendency to accumulate mutations, but because there is a very important tumor suppressor missing. And that is why, because they don't have a very high mutation load. Mutation load simply means that as the cells divide, they accumulate more and more and more mutations in subsequent daughter cell population. That is not the case with pediatric cancer such as retinoblastoma or some forms of lymphoma. And that's why they're very easy to treat given the right doctors and clinical access. They can be very easily treated because the background tumor load is next to negligible. Does that answer your question, Dr. Aziz?
Yes, absolutely. Thank you.
Thank you. Yeah, that was a brilliant question. So you can see here, someone has to be really, really, really unlucky for having cells which are able to move out from this to this place and manifest itself into cancer like this. Now, I want to highlight something. As you can see, myself hail from India. Is these adverts? This I'm going off the topic, but think about people trying to make their skin color fairer. Who is? Do you know who gets melanomas in UK? People with my skin color don't get melanoma at all. It's a natural protection. What people are trying to do is becoming more fair in color, and this is actually a societal problem. We should be proud of what color we have, and in fact, the darker we are, the chances of having melanoma through the sun rays is minimal compared to the white population. So we should be content and grateful actually that we are born with this color. Many of us, it's very common. The more south you go, the color changes. Should not impact anything, but what we should avoid is all these skin penetrating chemicals. Cortisone was what is there in these chemicals, which can lead to cancer, which is not yet studied. So coming back to the topic, you can see here these cancer cells need to cross a lot of barriers. The more they divide, the more they can become fitter. And as a result, what happens is cancers where the cell division number inherently is much more higher, there is a very high lifetime risk. And that's why you can see here, in order to reach this number where the cancer cells are actually gone through a lots of cell division, it requires time, meaning patients need to become aged. And as you can see from this chart here, the incidence of cancers actually don't increase tremendously until we are old. And why this? Because it allows us to buy the time to accumulate mutations in the daughter cells to carry out this journey, and that I'm showing you that somebody has to be really, really unfortunate to have a cancer diagnosis. What we are doing on top of our natural system, those mummies develop cancer, right? I showed you yesterday. So one way or the other, we will pass. We will never be. This is not the permanent home we have, right? The word. So what we are trying to do by exposing ourselves to these chemicals, cigarette smoke, alcohol, is accelerating this process coming from here to here, which is very sad, in fact. And the paper I was showing you, this Nature paper, which has the air quality index shown, would you be surprised? This paper shows in one of the diagrams that the findings that these cells which are exposed to the pollutants actually divide much faster. That's a bad news for a cell which has been left with mutations to divide faster, because then it is going to accumulate mutations at a much faster rate than a normal epithelial cells or any other cell type given. So that is sad news. So here we are. I think I have covered this. Dr. Aziz, are there any questions? I'm happy to take questions, or I can carry on. I don't want knowledge overload, so it becomes less interesting to each one of us.
Chat now, but their microphones are running on, so they can speak.
Yeah, if you have questions, please feel free to ask.
So yeah, your microphones are on, so please go ahead.
Yusra is asking, can we provide the study notes for today's and yesterday? Yes, Yusra, I will send it to Dr. Aziz. The notes from the yesterday are ready, and the ones from.
Dr. Asim, I already sent you the
Sorry.
Google folder. So you can just
Yeah,
upload
oh,
it.
that's correct. I will
Just upload them
upload it there. Yes, I will do that.
online as well, and we'll provide you the link so you can access it anytime.
Great. I have to say this is a superb initiative by Government of India to have programs like this so that we can all learn together. Brilliant ways to learn. And I hope by the time our lectures, both Dr. Aziz's and mine, will finish at the end of this week, you would have made your mind, is this something that you want to pursue as your career, research on cancer? And definitely if you don't want to do that. So, okay, I'm not seeing anyone asking questions. Are you happy for me to carry on, Dr. Aziz? And then I can stop. So you can see here, this is a model of cancer. Well, the question just came, so I'll stop and take. Sandeepan is asking a question. Can modern gene editing techniques change the fate of these mutating cells with age and prevent cancer? Sandeepan, that's an excellent question. Are you a faculty or are you a student? Please let me know in the chat box. Sandeepan is asking a very good question. Can we change, meaning can we reverse those mutations and make a cancer cell a non-cancer cell? Sandeepan, theoretically 100% possible, but to implement this even in a laboratory setting, to do it in vitro in lab, it will be difficult. Changes, there will be different changes, like Dr. Aziz was showing yesterday. Some people think they're sitting on an elephant, they think this trunk, those ears, they are the cancer. Other things that these pillars are cancer. First of all, what are the driver mutations? We need to know those driver mutations. Oh, you're a master's student. Excellent. Congratulations on your knowledge and your understanding, Sandeepan. So coming back to the topic of your question you're asking, you need to know what are the driver mutations. Some mutations are passenger mutations. They're just there because the pathway to repair them is not there. So how many of them you're going to change? You know the cost of gene editing, how expensive this is going to be. It is going to be enormous effort for any health economy, especially health economy in India, to do anything with gene editing for cancers which have very high mutation burden. Yes, something you can do for diseases like retinoblastoma, where you know exactly which gene to revert back to its wild type phenotype. So that makes a perfect sense for cancers where there will be less mutational load, to be able to change at the level of, at the embryonic level, to make those changes. But for diseases like this, when an individual has been smoking for 40 years straight and they have enormous changes to their DNA, reverting them back each time doing CRISPR, you know more than me, Sandeepan, cancer cells are not usually diploid. They have tetraploidy, they may have hypertetraploidy. So how many alleles are you going to revert back to their wild type phenotype? And we're talking about if not billions, millions of pounds to be spent. And again, you're treating a body which has already been exposed to a lot of different threats. So I would say this will not be viable. Giving you an example, gene therapy, people thought when it came, oh, this is going to be super viable. But now, many people think that gene therapy is not something which can be rolled out to masses of population. Having said that, there are hopes for genes where we know exactly what mutations are there, which can predispose. So if you can make interventions before cancer happens, for example, mutations in BRCA, which we very well know, might predispose patients having those mutations for specific forms of breast and prostate and other cancers, you can certainly do those interventions. So brilliant question. I have seen another question. And Dr. Aziz, if you want to add something, please feel free to do that to the answer I gave.
I had a more optimistic approach, Sandeepan, because we are already seeing the stories coming in, like sickle cell anemia was successfully cured, and even in the rare diseases, we are finding this because rare diseases are
That's correct.
really difficult to
I,
treat. So people are going for this,
I,
and
I
as
totally agree.
but yes, economy is one aspect that we need to definitely consider. Plus, in cancer, it's multifactorial. Coming back to the same point that it's not one mutation, except for there are some exceptions. Mostly, it's multifactorial, and it's a
That's right.
moving target. So you diagnose it today, you go after 3-4 months, you have got the target has moved. So that's why
That's
it's not
right.
going to be very quick in cancer. Thank you.
No, I totally agree. And on top, for diseases which are non-cancer, for example, thalassemia and so on, you know exactly what is the mutation causing these sickle cell-shaped RBCs to form. And in those cases, certainly you can do such interventions. That's much more straightforward than we're talking about cancer. You can see there are enormous mutation load in most of these cancers, which will be a nightmare for any pharmaceutical industry to use gene editing. Uh, I'm not talking about when I said I'm not pessimistic, but what I'm saying is the best disease treatment we have for prostate cancer, advanced disease, is a drug which has been used, sold to Johnson and Johnson, I think, for $8 billion, $8 billion, and it gives 3 to 6, 7 months to live. Now, I asked the question who discovered this drug in Memorial Sloan Kettering. I was in a talk and he was presenting. And I asked him this question more than 15 years ago, that why would I want to live? These patients who have this advanced disease, the cancer has gone into bones. Why do you want to keep me alive when I have such excruciating pain? The disease is really terrible. It cracks open the bones, causing enormous amount of pain in bones. And his answer was, well, some people can see their grandsons, granddaughters graduate from high school. They want to live. They don't want to die, basically. That's what he was saying. So even if it can at 3 months, the US government is happy. The insurers are happy to spend millions of dollars to treat one single patient. That was his response. So it is all in the perspective. I told him, if I have this disease, I would rather not want to be alive and give more trouble to myself and my family for another 3 months. So anyway, coming back to the questions that are in the chat box, Dr. Aziz, feel free to take any of these. I'm going to read them. Any specific reason Khadija is asking, prostate cancer chances increase after 40 years? One of the things that we have discussed, Khadija, is the rate and the time a cell has undergone cell division. So if cells undergo more division, you are older, your cells are more old, and the chances that they have accumulated more cell divisions increase, but also the time that you have been exposed to agents, cigarette, alcohol, those creams, cortisone knows, as I said, what is in there. Is there anything causing cancer? Then certainly chances of this increase as well. Zaid is asking what happens to cancer incidence with increasing age. As we discussed, they go up, as I mentioned, with age for almost every cancer. This is the same trend. Mustafa is asking, will these interventions, treatment therapy, will be more expensive like the mainstream? Yes, that is the problem, Mustafa. These drugs, which we can make for pennies, are being sold because they have patents. They're being sold for much higher price. Then certainly doing CRISPR using gene editing, the treatment will be super expensive on an individual basis. We already do treatments for CAR T cell therapies. Many of them are not even permitted, even in UK. As you know, there is no private health system as such, and the local population relies on government hospitals for treatment. Many of them are not really available to us because the evidence is very little that they increase lifespan, a healthy lifespan. Aparna is asking, can we not use stem cell therapy when diagnosed at the first stage of cancer? When diagnosed at this first stage of cancer, benign cancer, well, stem cell therapies, as you know, are used. You deplete from the bone marrow the cancer cells and then replant populated cells which you grow in culture. you can make those interventions. And yes, that saves life. Certainly, that's, I don't see any issue with that. So, Aparna, yes, but to roll it out in a big population and make sure that, developing a drug and then following it up through a longitudinal follow-up, 10 years, 15 years, it takes time. And many of the treatments we have, unfortunately, don't really stand the test of time. Many times these treatments may have severe adverse effect, which will kill a clinical trial of a drug, causing lots of closures of companies. So that's not a good news, of course. But yes, that is one aspect which has become a treatment in specific cancer cases, certainly. So we have 7 minutes, and I'm trying to see what I can complete. So on this slide, what you're seeing is again the same thing. The cancer cells is becoming fitter. This is called multi-hit model of cancer induction, which suggests that cancer actually does not happen overnight, and it takes a long time. And why it takes a long time? Because the cells have to be really, really lucky. Cancer cells, I meant, they need to be really, really lucky to have these first mutations, which will give them growth advantage. Second cell mutations may give them advantage to grow more uncontrollably, or may be able to, for example, other mutations may be able to give them more chances to move inside the tissue, for example, invade in the without mutations. And that need mutations that will give them that quality, that ability, that they're able to migrate. There may be fourth mutation. So this is oversimplification. Don't think there are just 4 mutations. And the fourth mutation will make sure that they can escape into the bloodstream and survive. There are enormous levels of mutation in a cancer cell. Sometimes not just one gene mutation, multiple genes mutated. And as Dr. Aziz was telling yesterday, pathways are important. There may be whole pathways switched on or switched off, which will allow them to survive in their journey when they're going from the primary organ to sit and metastasize to a different organ through blood. This journey may take very, very long. Now that we have epidemiological data, we have data from lots of patients, especially in Western countries. We now know some hotspot mutations. We know TP53 is a good guy, is a good gene which has lost its function in. We will see that in the next lectures. Lost its function and become actually causes cells which have the mutations to become cancer. And this is one of the examples to follow this journey where we have first, second, third, and fourth mutations. Will be a cancer which Dr. Aziz's lab studies. It's called colorectal cancer, and we know very well defined stages where cancers can actually show different abilities to attract and progress towards their metastasis journny. So starting with the polyp and might take Dr. Aziz 20 years for cancer to metastasize to viscera and so on, and each of them may be accompanied with different set of mutations. In some examples we know them, in many other examples we don't know them. So if I ask you this question, DNA repair, is it good or bad? You can think about this in your own terms. Is DNA repair good? If you're a normal human being, yes, 100% we need good DNA repair system. There are people who have, on an average, have lives in the world over 100 years. Talking about some parts of North India, extreme up north, Ladakh, and so on, and North Pakistan. People live as a norm 100 years plus. May have very good genes, and when I say good genes, maybe a very efficient DNA repair system, coupled with, of course, good air quality, smoking seen as a bad thing, as bad societal trait, and they might have a very good chance to live. And of course, DNA repair is good, but DNA repair is bad for cancer cells, and we will see this in the subsequent lectures. So, Dr. Aziz, are you?
Well, I can tell you one positive aspect
Sure.
about loss of DNA repair.
Yes, please.
When using the radiosensitizers like iododioxyuridine, this
Yeah.
is a radiosensitizer which is helpful for radiation therapy.
That's
So
correct.
people who are mismatch deficient, they respond better to these therapies and these radiosensitizers. So why? Because their mismatch repair status is lost, and they
That's
can
right.
retain more of this, and then they get better response. Second part, in the immunotherapy against checkpoint inhibitors, it's well known that if you have got mismatch repair deficiency, you will be able to generate better epitopes for
That's correct.
the success of the immunotherapy. Otherwise, I mean, you don't even respond. So now it's a standard that somebody who is DNA repair deficient will be given the checkpoint inhibitors, and then those who are proficient will not be given
That's
these
right.
check.
Microsatellite instability.
Yeah.
That's absolutely right. Yeah, absolutely. These are all great points. Certainly makes a lot of sense. So we are already heading, as Dr. Aziz was saying yesterday, towards personalized treatment um, based on the tumor characteristics and how good we are to differentiate between these tumor qualities from one individual to other at a population scale. So, Dr. Aziz, would you like me to carry on with the rest of the talk and then the tutorial?
Yes, I think 4 to 5 was your tutorial schedule. So we can take a short break if you want.
Okay, yeah. Let's meet in.
So we have been speaking. And meanwhile, the participants can put their questions in. We'll take a short break and then we'll resume.
10 minutes, Dr. Aziz, is fine.
Yeah. Yeah.
I'll leave the slide.
So I have posted the link for all the lectures, the Google Drive. So the participants can look at this and they can find my lectures there. I've already uploaded. And I would request Swati to please upload today's videos as well in my folder. And I hope Dr. Asim will be putting his lectures in the folder as well.
I will do that. Thank you.
We have real-time access to all these materials that we are talking
I'm
about.
always running behind, so I have this lecture to cover before the tutorial, and that's why I leave it until the last minute because I never finish my lectures on time. I get so taken by my lectures.
Totally understand. So, and let's,
I-
there are about 59 participants, but please make this session vibrant, asking questions, discussing. This is
yeah,
what we need.
it should be interactive, really, both ways because what I do tell my students is use this as an opportunity to, you know, get rid of your, if you're very shy, if you have any inhibitions to talk in public, and use this as an opportunity to prepare yourself for any upcoming interviews, for example, that would certainly help.
Yes, because see, there's no question of that question. So just ask.
That's right.
It's not like I don't know it, everybody else knows. No, come out of that inhibition and ask. Even if you are asking why DNA is having AGTC or AGUC, it's fine. Not a bad
100%.
question. So once again, I would encourage and request and exhort you all to ask as many questions as you can. Give us a hard time.
Please do. I hope everyone is back and we can start.
Just there's a slight drop in the number, so it was yeah, 10-15 people. They just went on a break, longer one.
Okay,
But
we are
yes,
recording this anyway, right?
we can start. Yes.
Okay, so yeah, let's just start where we stopped. Good. So I'm not going to go into the details, and this is relevant to the repair systems, and that's the reason I'm showing you this slide. What you can appreciate is that the 5' end of a nucleotide chain is always a phosphate you will find, and this is connected with a phosphate ester on both sides, meaning it's a phosphodiester bond uh, through the another nucleotide. The 3' hydroxyl is where the new nucleotide, incoming nucleotide, comes. I believe you all know this. What you also know here is that this bond is called the glycosidic bond that connects the base, the nitrogenous base, with the pentose sugar. Loud and clear, I'm sure everyone knows this. And then there is this hydroxyl at the 3' end, which acts as a substrate for the growing polynucleotide chain. I'm sorry. And there are repair systems, of course, that are needed in the body to correct these defects that happen to the DNA. What I was telling you about the damage to the DNA, there were 3 prominent ones. So the oxidative stress-induced oxidative damages. There was second one, which was the hydrolysis of nitrogenous bases, specifically purine hydrolysis, and the third one was the deamination. So we have discussed these 3. All of this, you would see that into the repair process, they will lead to generation of these single strand breaks. Do we have to worry about single strand breaks? Not really, because we have efficient systems to repair single strand breaks as well as double strand breaks. The only difference comes about is that single strand breaks are very well tolerated by the cell. Plenty of single strand breaks are generated in a cell all the time. What is not tolerated by a cell, meaning a cell will not be able to continue its normal life cycle, is if they have a double strand break. Would you believe that as little as 3-4 double strand breaks are sufficient to kill a cell? If it is not repaired, the cell will die. And that is in terms of normal cells. If the cells are not really cancer cells, they cannot sustain life if they have any unrepaired double strand breaks. And the way these radiotherapeutic treatments, anti-cancer drugs, cytotoxic drugs function is by destroying the DNA. We have pathways which can correct these double strand breaks. In particular, this homologous recombination pathway, which we will try and cover in the tutorial if we get to start and finish the tutorial today. If not, no big deal. We can catch it up tomorrow at the same point. But the whole essence of this is to cover things which you will find very hard to get in any books or research articles. And that's why I'm taking my time to allow you to understand this. If some of you understand and not everyone, I'll be more than happy, of course, if you understand all of it. Students I'm talking to, but yes, please let me know if there is something which is not sure in your mind. So what happens here, these are some enzymes that act on a double strand break. For example, you see these enzymes which will cut the phosphodiester bond and leave a nick in the DNA, as shown by this scissors. And this is caused by an enzyme called endonuclease. There are enzymes working to oppose this function, and they can seal this nick back. And these are very well known as ligases. I'm not going to go into the details. You can read that very easily in any books of molecular biology. And these are some textbook enzymes. The only intent I have is to show you how these things actually work when it comes to the DNA repair, especially in context of cancer. You have enzymes which start cutting not from the inside but from the edges of the DNA. We call them exonucleases enzymes, and these three are very important in the integrity of the DNA, especially these ligases, to make sure any nicks are any created during the repair process, because eventually you will create these nicks as you start to repair the DNA. And then these enzymes are there to take care of any defects. So the first part is this enzyme, DNA polymerase. You can read all of this information if you like in your own time. I'm going to focus on the crux of this slide in the interest of time, but also, as I said, this is something of very relevance to this talk. So what happens is DNA is synthesized in 5' to 3' direction. We all know that 5' phosphate to 3'. And this is the ability of the enzyme to carry out this function, this polymerase. But they also have a different function in opposite direction, meaning 3', not from 5' to 3'. They act as a polymerase, but in the other direction, they act as exonuclease, you can say. And meaning here, what is that they can remove any nucleotide which is wrongly incorporated. So if you see this as a template DNA, this is the new strand being formed. And what you see here, the enzyme, unfortunately, glues a wrong nucleotide. There should have been a T. Is there a T here? No, there is a C. It's become part of the DNA. It is removed under normal circumstances. So if you have these enzymes, most of them, almost 99% of them are proteins. We know that very well. What happens to the protein coding genes? If those genes are mutated, these enzymes are going to lose their ability to correct these, identify and correct these situations where a new wrong nucleotide has been put. So this function is called proofreading function, and sometimes the DNA polymerase will not have this proofreading function thanks to the mutations in the very enzyme, which was meant to repair that DNA, and that will leave some mismatches behind. You can see what happens when you have these mismatches. These tumors will have, because these DNA polymerase is now mutated, they will have super high mutation rates compared to normal cells. So that is one of the initial things happening. Cells which are not yet abnormally dividing, will be very hard for us to identify those cells. There may be just few cells in a big organ which have started to have very high mutation rate, thanks to the mutations in enzymatic activities such as this enzyme, just proofreading activity of this enzyme. And because of this, they will have sometimes new proteins being formed. Why? Because if you have mutations, these may be mismatch repairs. Sometimes what will happen, if they are not repaired, they will give rise to proteins where some amino acids may be different. And as a result of this, the codons will change, amino acid will change, some amino acids. And as a result of this, the protein will change as well. How many proteins? Well, a lot of them. So they will have a hypermutated phenotype, these cells. They will make new antigens, meaning with regards to the immune system. As Dr. Aziz was telling, these are the cells which will be an excellent candidate for immunotherapy if they were to become cancer. So first question first, how our body repairs them. There is a pathway, and you can see any good textbook that will talk about these mismatch repair pathway. This is called mismatch excision repair, which needs these enzymatic pairs, these proteins, MSH2 and MLH2 and MSH6. They are needed in order to carry out this repair function. I'm not going to go into the details of it, but what happens if this mismatch cannot be corrected? Have a look here, what is happening here. In patients who have lost the good copies of these proteins, which carry out this particular pathway to repair the mismatches, you can see there is a mismatch, AC, and this has been now corrected as a result of this. But what if we don't have this good copy of these enzymes carrying out this function, it can actually lead to lots of different diseases, including this type of polyposis, HNPCC cancer. And you can see from the endoscope, the cancer is quite obvious. If you take out these tissues and do histological analysis, you can see here, this is a histology of this colon tissue, and you can see these are nice colon cells sitting here next to each other. These are all, of course, sadly cancer cells. But what is remarkable is that there is an infiltration of the lymphocytes. These are the cells which are trying to attack these tumors and kill them. That is exactly what Dr. Aziz was just mentioning, that these cells which have these mutations, which they cannot correct, will lead to a lots of new antigens being formed, meaning new proteins which do not exist in normal cells. And as a result, they will invoke an immune attack, as you can see from this slide. What you can also do is that we were talking about this deamination, which is spontaneous. We have repair pathways in our body which can take care of this. It's called base excision repair, which can actually revert back any deaminated DNA into a wild type DNA. There is a whole process of it. As I said, I'm not going to go. This is at best MSc first semester stuff that you can study. And this is the last pathway, nucleotide excision repair, which becomes active when there are bulky adducts formed in the DNA. And the function of this is very simple. The nucleotide excision repair proteins would repair any bulky adducts which form, and as a result, they distort the structure of DNA. So when I was talking to you about the BPDE, what it was doing, caused by benzopyrene exposure, it is creating a bulky adduct. Well, these are thymine dimers. Again, I'm not going to go into the details. Exactly doing the same thing. They're causing the bulky adducts to form, triggering this NER, or nucleotide excision repair system, as shown in this slide, taken from, I think this is from one of the textbooks. I gave you the link yesterday. So what happens if this system fails? You have illnesses like skin diseases, xeroderma pigmentosum, which can make these patients more prone to developing different types of skin cancers and not having these. So nucleotide excision repair is very important. It keeps our skin safe from harmful rays of sunlight, so that we don't develop illnesses, sadly, as shown on this slide. But nucleotide excision repair could be bad when we are treating cancer patients. Have a look at this review, and this shows exactly the same thing. So one of the chemotherapeutic drugs, which is used, I would say, mostly to treat a lot of different cancers and has been very successful, is cisplatin. And you can see here there are evidences, irrefutable evidences, that patients in which cisplatin initially works, but then it fails to work, it turns out that the nucleotide excision repair becomes activated. And do you want that repair system to be activated? No, you're trying to kill those cancer cells by damaging their DNA beyond repair. So activating an excision DNA repair pathway would be a disaster for that individual undergoing cisplatin therapy, because cisplatin will stop to function. So these are some of the points which I have summarized what we have discussed. I will try and now jump on to the tutorial today, which is harnessing the tumor defects for its own demise. Am I audible, everyone? Dr. Aziz, can you hear me? Perfect, Swati. Okay, so let's crack on with this tutorial. So we discussed, I was showing you those migratory birds and so on, that these, the cancer cells are very smart. They have undergone extensive training, but in doing so, they actually make themselves in some ways more vulnerable. Shall I say? In other words, they have some tumor defects. And because of these defects, if we can identify those defects, we can kill them. And you might have seen, if you visited London, these scaffolding sometimes in around the Big Ben and the Parliament in London. These scaffoldings as a part of restoration. Well, this scaffolding is not restricted to buildings and structures, but also to the DNA. So the previous lecture I was showing you just now, these nucleotide excision repair pathway, base excision repair, I touched very briefly, and mismatch excision repair. Many of these pathways actually need this molecule called PARP. So PARP is very important molecule, and its role is very well known in single strand break repair. You would say, hang on a minute, you are saying single strand breaks are taken care of in the body. It is double strand breaks we need to worry about. That's correct. But single strand breaks, I also said, if they are not really taken care of, they can convert into a double strand break. So PAR function is to make sure any single strand break is not converted into a double strand break. You can see what it does. At single strand breaks, PARP actually makes a polymer of itself. And in doing so, it actually creates a scaffolding to recruit other proteins, other DNA repair proteins, which are needed for the repair of DNA. So all these pathways I just showed you, these nucleotide excision repair, mismatch excision repair, the base excision repair, all of them will have some role, and they will need part one to start this scaffolding. You need that scaffolding to recruit these proteins. So that is needed. I've shown this slide already. So this one already. So what happens here is that there are proteins which carry out repair of DNA double strand breaks, because we cannot really rely on PARP. It is one of the, if you see any immunohistochemistry, immunocytochemistry slides, you would see that one of the most frequently abundant protein in cell nucleus is none other than PARP. It is very rich, the cells in PARP. You can see what is happening here is that there are proteins which are there to repair the DNA double strand breaks. Where do you get double strand breaks? Well, radiation, treatment, cytotoxic treatments, platinum drugs, all lead to double strand break formation. We know that very well. But sometimes if single strand breaks are there and they are not repaired, they can become double strand break in daughter cells. We know that as well. So what happens here is that if they are unrepaired, they will become double stranded. And this double strand having inside DNA two phosphodiester bonds broken in opposite strands will trigger a response called the double strand break repair response. And this response is coordinated by none other than BRCA protein, which I was telling you yesterday is a growth protein, but it is degraded, it is removed by the formaldehyde, which is produced. One of the things produces it is alcohol consumption. So what happens if you remove this? That the cells will not be able to carry out this entire pathway to repair the double strand break. So you need that pathway, BRCA pathway, to repair any double strand break, which is a product most of the time in cells when the single strand breaks cannot be repaired. So you have these other complexes like the single strand break repair pathways. I did not go into the details. I'm not going to go into the details of this pathway either, but I'm telling you more high level and giving you more high level information why this is important. And I hope you will be able to understand that. So what is happening here? Many of them will activate this kinase called ATM. ATM is a sensor of DNA double strand break. We all know that. And ATM is going to again amplify this DNA double strand break repair, in which the histones will be phosphorylated. We know that. And there are proteins called RAD will be recruited to the DNA, and this DNA will be repaired through homologous recombination. So the role of BRCA is very important in all of these pathways, the recruitment of these complexes, the ATM activation, and the recruitment of RAD51. BRCA is testing is done very regularly in many Western countries, even in Eastern countries, including India. And it has been noted that people with first degree relatives who died of cancer, such as breast cancers, who have BRCA mutations, their relatives, first degree relatives, will be much more susceptible to developing a BRCA related other cancers, because BRCA is a good gene that protects us. We discussed yesterday what degrades BRCA is alcohol can degrade BRCA. So cancers, many cancers will not have BRCA function, meaning if there are double strand breaks, they will not be able to uh, repair those double strand breaks because they need BRCA to repair double strand break. Does that make sense? So what happens is this is something which is dubbed as synthetic lethality. It's a complicated slide, but I'll try to make it easier. Imagine gene A is nothing but something that allows cells to take care of any single strand breaks. You have single strand breaks, and gene B is like gene BRCA, whose function is to repair any DNA double strand breaks. So you have single strand breaks, you cannot correct single strand breaks. Then you have gene BRCA or B, which can correct the conversion of any single strand breaks into double strand breaks. Both pathways are important here. But what happens if gene A function is gone as a result of mutation? Meaning, if you have single strand breaks, they will be converted into double strand breaks. But double strand breaks will be taken care of because gene B of BRCA is intact. The cells will stay alive. This is what is happening in cancer cells. This is what is happening in normal cells. Normal cells have repair systems to repair single strand breaks, but they also have systems to repair double strand breaks. Cancer cells may not have the system to repair single strand breaks, allowing cancer cells to convert any single strand break into a double strand break. And double strand breaks, if they have it, BRCA may be there, and that can help. What happens in women with breast cancer is that BRCA function, as I mentioned on the last slide, the BRCA function is not there, but they might have single strand break repair system, which is still intact. As a result, cancer cells will still survive. Why? Because single strand breaks will not be converted into the replication cycle into a double strand break. If there is no double strand break, you don't need BRCA. So BRCA is a start because many women are not able to convert to repair their DNA double strand breaks. These cells essentially should have died, but they're not dying despite having double strand breaks. So they are already becoming fitter than normal cells who need to repair their double strand breaks. These cells don't. So what happens in synthetic lethality is, let's say BRCA gene function is off in many women, you can make inhibitor to block any single strand break being repaired. So if you block this, what will happen in cancer cells? That they will die. And that is exactly what is shown in this next slide. What you can see here is that there are proteins like PARP inhibitors. One of the examples will be olaparib, which is a clinical drug used across the world to treat many forms of ovarian, breast, and prostate cancers. Now, and you can see how does it function. If DNA has any single strand break, which I discussed with you, is happening all the time, we generate spontaneously single strand breaks in the DNA as a result of the repair process. Any of those pathways, mismatch repair, base excision repair, nucleotide excision repair. If you go and have a look, revisit those slides or your books, you will find there are always some part of the pathway which generates single strand breaks. And what is happening here? There are single strand breaks. PARP is there, forms a scaffolding, and then the cells will never have these double strand breaks. But what if you block PARP function? If you block PARP function, then these single strand breaks can be converted into double strand break. Okay, very good. What happens then? It turns out that PARP inhibitors don't really work on normal cells. They don't kill them. Why? Because normal cells will be able to have their double strand breaks repaired through the homologous recombination pathway, thanks to the BRCA which they have. So normal women, the breast cells, epithelial cells will have a BRCA. Even if you block their single strand break repair system, which will convert the DNA into a double strand break containing DNA that can be repaired by BRCA. So what about women who don't have? Many of the women have germline loss of BRCA function. They don't have any BRCA. And as a result, what happens here that these women, when treated with inhibitors of PARP, their tumors will die. Their tumors, the cells will die because they are not able to repair the double strand break for which they needed BRCA, and that is absent. So you can actually have development of a therapy by which you can convert any single strand break, which are normally non-significant. They are generated in their millions in every cell all the time. You can make that into a double strand break. Normal cells will not be affected because they have a system, BRCA, thanks to BRCA, that they can repair these double strand breaks. The cells which give rise to cancer as a result of having BRCA deficiency now, because they don't have a good copy, because they're defective in repair of double strand breaks, you can make them more vulnerable to PARP inhibitors. And that is what is called synthetic lethality to treat breast cancer, women with breast cancer using drugs like olaparib, which are PARP inhibitors. Uh, so basically, you can imagine this situation is like this. PARP inhibitors normally don't do anything to normal cells who have an intact double strand break repair, homologous repair system in place. But in women who don't have BRCA, what will happen? The cancer originated because there was no BRCA in the first place. So what will happen? Any single strand breaks which are being generated by normal cells and cancer cells alike all the time, those single strand breaks will become lethal as a double strand break. So you can imagine this as a barbecue causing a wildfire in a Californian desert, causing cancer to be treated. And this is an example of synthetic lethality in breast cancer. You can see there are some cells in breast. Not every epithelial cell is a cancer cell. You can see there are also normal cells, and normal cells have a good copy of BRCA repaired pathway gene. And you can see here any single strand break, which is happening, as I said, all the time. PARP inhibitor will convert them when taken orally. It's a capsule. So every cell will be exposed to PARP inhibitor. Sorry. So what will happen here is that any single strand break will be converted into a double strand break. But normal cells have good copies of BRCA gene, and they will allow this double strand break to be corrected, and the normal breast cells will survive. What happens if the tumors if they're heterozygous for BRCA, meaning they have a good copy and a bad copy of BRCA? These cells will actually form resistant tumor cells because they are pretty much like these cells here, where the PARP inhibitor can convert single strand break into a double strand break. But because of the virtue that they have a good copy of BRCA, they're not going to die. They will become resistant to PARP inhibitor olaparib. What is going to happen over time? One of the things happen in cancer is the loss of heterozygosity. As a result, what will happen? Some breast cells shown here in this dark purple is that there will be loss of both copies of BRCA. So they don't have any system to repair any double strand break like this situation or like in this situation. And as a result, what will happen? You will have single strand breaks all the time. As I said, PARP inhibitor is going to convert it into a double strand break. In normal cells or in resistant cells, this will be corrected by BRCA. These cells have no good copy of BRCA, and as a result, these breast cells, which will be the most dangerous ones, having lost both BRCA copies, will eventually die. And you can see here, this therapy will only work if there are no good copies of BRCA. Even if normal cells who have a good copy of BRCA or both good copies, they will not be affected, meaning there will be less side effects of PARP-based therapies. So you can see here what we are doing with aldehydes, formaldehyde, alcohol consumption. We are decreasing the level of BRCA gene. So there may be good copies of the gene present, but you may not have sufficient protein itself. So there is no change at gene level, but there is less protein, meaning what will happen in the first place? These cells might actually become cancerous. And once they become cancerous, then you can see the chances are enormous for them to kill this patient. So first of all, there are changes happening at germline as well as somatic mutations. On top, the environment is creating the deficiency of good genes, in this case by formaldehyde, which can lead to basically breast cancer. And it is known women who drink are more prone to developing uh, forms of breast cancer non-existent in non-drinkers. So what we did in the lab is, this is a paper we found that there is a similar relationship between the not BRCA and PAR, but PAR pathway and androgen receptor. I'll tell you in the next lecture what androgen receptor is, but have a look, this double strand break repair pathway I was showing you before. Like BRCA in breast cancer, androgen receptor repairs DNA double strand breaks in prostate cancer cells. So, okay, so BRCA is not there. Yes, BRCA is needed, but androgen receptor, what we found is needed to carry out this double strand break repair pathway. So what will happen? Nature is the smartest guru we have. So what happens if you block, and this is seen in breast cancer as well, if you block this androgen receptor, you see what happens here. This is the slide from a patient who were given therapies to block their androgen receptor pathway, which, by the way, is a regular standard of care treatment, block their androgen receptor signaling axis. And you can see this is the biopsy before androgen receptor blocking therapies and after androgen receptor blocking therapies. Can you see what is happening to the PARP activity? So PAR is the moiety which is added to the DNA by PARP enzyme. You can see here PARP enzyme becomes super active after we have blocked the androgen receptor signaling axis. So you have enormously high level of PARP activity. Normally, the PARP activity is very, very low. But when you block androgen receptor, the PARP activity goes up. What is the rationale for this? Well, that's a perfect rationale, because if system realizes, if nature sees that they don't have a DNA double strand break repair pathway, why not enhance single strand break repair? So if you can use PARP, you can enhance the PARP activation, you can probably inhibit the single strand break converting into double strand break in the first place. And that is exactly what happens in nature. When the cells realize they cannot afford to have any double strand break, they will upregulate the single strand break repair pathway so that there won't be any single strand breaks. And in the S phase of cell cycle, they will therefore not be converted into a double strand break. So what happens to treatments of tumors with inhibitors of PARP and inhibitors of androgen receptor? Have a look in this tumor xenograft model, and what it is showing you on y-axis is the tumor volume versus the time of incubation when the drug was given. You can see this is prostate cancer cells. They grow very well in mice. What happens with olaparib, which is a PARP inhibitor? Basically nothing. Why? It is not toxic to cells, because single strand breaks will be converted into a double strand break by olaparib like drugs, which is a PARP inhibitor. It doesn't matter because androgen receptor is still active in them. So no effect on tumors. What happens if you give androgen receptor inhibitor bicalutamide? Well, very little or no effect at all actually on tumors. This is not significant. Meaning, and if you block androgen receptor, it doesn't matter. I'm sorry about that. You can see what is happening here. If you block, sorry, the drug androgen receptor inhibitor, what happens? There is no effect at all. Why? Because the single strand break will be repaired by PARP pathway as we saw here, but it is only when you block both of them. So you do have a single strand break repair pathway compromised in this group. Many of them will have double strand breaks, and these double strand breaks don't have the androgen receptor pathway intact. As a result, this will be converted into a double strand break, and those cells will then die. So this paper, we discovered this in over 15 years ago, published this in 2017. It was covered by print media. There were trials done on this, and based on this is a recent Nature Medicine paper, which actually shows what we showed in mice, that there is an effect, there is a benefit of blocking both PARP inhibitor, blocking PARP pathway and the androgen receptor pathway, and the combination can lead to more tumor death. They use this treatment, combination treatment, and actually found that this is actually very effective in treating patients who have advanced prostate cancer, in which a combination of this gives them a prolonged life. And they did thankfully quoted our paper, and they've received FDA approval of this therapy, which is now used throughout the world. I'm pleased to say, for the treatment of advanced prostate cancer, combination of PARP inhibitor, olaparib, and other drugs which inhibit PARP, in addition to androgen receptor inhibitors such as abiraterone. So thank you very much. I'll stop with this and happy to take any questions for this tutorial. We have 14 minutes for any questions that you would like to ask me. So Swati, I'm not sure if you have activated the mics that they can ask me questions, the participants. Um, I'm more than happy for them to type their questions and I can try to answer. Although my voice, as you can see, is not very good these days, but I hope I'm understandable. Thank you. Thank you. Thank you. Dr. Aziz, any questions, any advice, any feedback, or from anyone else? Sure. So thank you, everyone. We will. Thank you, Swati. Let's reconvene tomorrow. Oh, there is a question. There is a question actually. So Happy is asking a question. Currently, my third year of BSc Biotechnology. So I do pursue a career in precision medicine. Although this course is primarily designed for master level higher, therefore, utilizes in the field, I would get you an insight on how to undergraduate can strategically prepare. I think, Hafi, what you need to do, first of all, I was in a similar dilemma when I did my master's in Jamia Hamdard. I first and foremost tried, and during my master, I thank God I cleared CSIR, and they started back then ICMR. That was the first year. So I qualified those. I took admission in JNU. I qualified their exam as well. It would have taken. They had facilities, but I realized it would have taken 5-7 years at least to complete the PhD. That was not something I was hoping for, and there were issues. Of course, I'm talking about 2003. Actually, yeah, 2001 actually. So what I would suggest is to do some substantial training, let's say 1 year training in a good research lab, and there are plenty of such labs in India. Make sure you're able to make some contribution to the research and publish research articles. So I understand most of you are super bright students and will graduate in your BSc or MSc with the first class. On top of this, what will attract future, you know, employers is your ability to demonstrate in your CV that you have actually meaningfully contributed to a publication. Have yourself as an author on the publication. This is what I've done. Believe me, I have second year BSc students in my lab. They 2 weeks ago finished writing a paper on which two of them appear as second authors, equal contribution. That is what I want students to do, but they can they time out 15 months from their conventional degree. So instead of 3 years, they have a 4-year degree. But I ensure that anyone who does that in my laboratory spend a lot of time, hard work, uh, pays off. And then as soon as they finish their BSc, they are on track to do PhD. Most students who I have in my laboratory now and previously never had any masters. They came right after their BSc, and having spent a year in a top lab in UK or in Europe elsewhere. Sadly, for me to take any students from India is difficult because the tuition fee for Indian students is about 25 lakhs per year. Uh, for UK students, it's cheaper. It's about 4 or 5 lakhs. What would be best is I was in the same situation when I was looking for PhD studentships, so I had to write my own grant to support myself, which was thankfully successful, and I joined a laboratory with which in Germany, with which I wrote a grant application. And the guy, my supervisor, was honest enough to see my potential, invited me on that grant to do my PhD, paid stipend. My tuition fees was about 100 euros, which is 10,000 rupees or something, compared to in UK, which is 25 lakhs. So the quality of education you get in Germany is very good. Having said that, if you're able to get any positions in UK which are Wellcome Trust funded, any PhD studentships which are Wellcome funded or Cancer Research UK funded, usually they cover the tuition fees for international students as well. So I hope that answers your question. Any summer internships opportunities. Safi, I don't sadly take any students, even local students who have no visa problem. I don't take anyone because it creates, to train students, it takes a very long time. And I don't have a very big lab. I have 4 people working in my lab at the moment. And we don't have very much time that we can spend on training short-term students. So sadly, I have to disappoint a lot of students. I decline any short-term request. I take students if they want to come at least for 12 months, 15 months. And any of them who've done this usually will get a good PhD studentship elsewhere, because they can produce evidence of having published paper when they're BSc students. So certainly a lot of employers, a lot of supervisors in different universities are interested in taking those bright students. And one of the criteria I have is that they should have very high first class marks so far in their degree, because otherwise I normally find it's a waste of their time and mine. Thank you. I hope, so Fatima is asking another question. She's pursuing chemical engineering from AMU, wish to pursue research in this field. What skills should I focus on, and how can I academically pursue it? I think, Fatima, certainly if you're interested in this area, one of the things which is currently a hot topic is medicinal chemistry. And of course, you see this click chemistry being used in designing drugs and so on. So certainly if you, chemistry, I have to accept research in chemistry. India is one of the, at the forefront of chemistry research. So certainly if you're able to do your research, publish some papers while you're doing masters, which is quite a possibility. You can look for PhD studentships. I have to say, in Europe, there is no tuition fees as such. On top, you get your stipend, which is many times, depending on how spendthrift you are. Normally, it is sufficient to keep you get by during your PhD years. But you have to realize you're not on a job. You're getting a degree and then getting paid. I love that aspect. I do something, cancer research, which is at the heart of my professional life, and I'm paid to do that job. So if you're able to publish some papers while you're doing chemical engineering degree there, certainly you would have much better chances, especially if it is in the area of medicinal chemistry. Of course, you know your field better. With I do collaborate with medicinal chemist, among others, and I used to know someone who was one of the persons working here. He was a graduate of, he did his PhD from AMU. He was a top scientist, medicinal chemist in one of the top pharmas. I've never not met him in last 10 years, but definitely there are much more scope for chemical engineers in medicinal chemistry area. Swati, are there any more questions or? Perfect, thank you. Thank you. Okay, we'll meet tomorrow then. Thank you. Take care, everyone. Thank you for organizing this, Swati and Dr. Aziz. Bye, everyone.